what we are going to talk about is rather than the membrane itself, because we know the composition of the membrane and how it is from the previous unit. Here we are talking about that how the proteins are transported through these endomembrane system in different destinations, and how outside things can come in a short part at the very end. So first slide that what we have is an overview of eukaryotic protein sorting or trafficking. This is a very important slide. So what you are looking at, that you have these mRNA right here in red, and then you have these round structures. These are the ribosome, which are made up of RNA and proteins, many of them, very large molecular weight. And on the top, you have a small subunit. At the bottom, you have a large subunit. And the tiny, small blue part that you are seeing is a growing polypeptide chain. So just to refresh your memory, that the mRNA will have some untranslated region, and then you will have a start codon, AUG. And as soon as the ribosome reads the AUG, it will put a methionine, the first amino acid, onto the protein, and it will read every 3 letters, which are called the codons, and it will add the corresponding amino acid from the pool of 20 amino acids, which are brought by the tRNAs, and the polypeptide chain will just add bond in between the C and the N terminal of the amino acids, and it will just form the polypeptide chain. As the mRNA, it kind of think about that it ratchets through the ribosomes, the polypeptide chain will grow. At the same time, what you also see that particularly in the bacteria, that many ribosomes can bind to the single mRNA. And this basically makes the protein synthesis much faster. So think about that. The first ribosome, this was the first ribosome on the very end. This is if you just follow the arrow, this was the first ribosome that was initially bound at the position 1. And then as the mRNA ratcheted through it, it just came here. This bound second, this bound third, this bound the last. And ultimately, this fourth one will come in this place as well. So once this ribosome, which is right here now, which is a considerable amount of polypeptide chain, will reach the stop codon, at that time, the mRNA, the large and the small and the large subunit of the ribosome and the protein will be released. They will dissociate. So the mRNA new ribosomes can bind and do the same process. But this is the important part, that this is, which is very important, is that this is essentially, so you have a start codon, you have a stop codon, and stop codon basically disassemble the system. So what is the take-home message from here is that in the cell, all these proteins that are synthesized do need an address to go to a destination. If it doesn't have any address or signal into them, and what is an address? It is just a sequence of particular amino acids, one after another, and that can be recognized by other proteins to take them to a certain destination. Just think about that. This is a letter, so that your protein is the letter. On top of that, you have an address delivered by the post office and the other people, and these are the supporting proteins to deliver that protein in a particular part of the cell. If it doesn't have any address, then it stays in the cytosol. So that is one of the most important part. And this is your question, that if we just ask a typical question, that in absence, very carefully listen to this, in absence of any signal or targeting sequence, where is the expected location of the protein? Remember that will be always the cytosol or the cytoplasm. Without a signal, a protein will remain in the cytosol. And there are many proteins, of course, hundreds and thousands of proteins in the cytosol, but without a signal, that is in here. See that this doesn't have, it's all blue, doesn't have any pink part. Now, if we look at the picture on the right-hand side, 1, 2, 3, 4, 5, 6, one after another, all of these proteins now have a targeting sequence, which is shown in the pink. If has an address which says that it needs to go to the mitochondria, it will go to the mitochondria. If it does have a signal which tells the other proteins to take it to the chloroplast, it will go to the chloroplast. If it's a signal that will take it to the peroxisome, it will go to the peroxisome. If it's a nuclear signal, it will be taken into the nucleus. So all mitochondria, chloroplast, peroxisome, nucleus will have their own unique sequence for protein trafficking. Now, this part is, we are not going to discuss this part because it will be similar to the one that we are discussing and extend the same thought. But for the purpose of the course, that it will be almost impossible to cover all of them. So we are going to cover this side of the trafficking. But again, remember that targeting to these things, that the signals are different, the proteins will be different, and some mechanisms could be a little bit different because some of them are double membrane. And other than that, we are going to talk, they are the same. So let's see that what is happening on the left-hand side. So the most important part is that we collectively call this pathway as a secretory pathway. And sometimes the name actually suggests that it is making protein which are secreted from the cell, and that is basically a misnomer. So do not get carried away with that. Secretory proteins essentially do not make proteins to be only secreted from the cells. It does, but there are several destinations, and make sure that you get that very, very clearly. So if I just write down that what you have, number one, that if you have these proteins, you have an ER signal sequence, and no matter what, all secretory proteins are going to be synthesized from the ER through the ER Golgi. Then it can go to the lysosome, it can go to the plasma membrane and secreted outside the cell. So now, all of these proteins may have two different components other than the secretory part. So think about that. In this pathway, whatever is synthesized, you have first a soluble type of protein. These soluble proteins will stay in the lumen, like these proteins are soluble proteins, which are in the lumen of the ER. On the other hand, there will be the second type of protein, which will be the membrane proteins. Many different types. Where are the membrane proteins? This is your membrane protein. I'm just drawing it in a different color. So you have a membrane protein here. You have a membrane protein here. You have a membrane protein here. So you have a soluble and membrane proteins. Now, in between these two, also, they have several destinations. Say, for an example, some of the proteins are enzymes which are soluble, and they are endoplasmic reticular enzymes. That means that they modify the proteins and work with them and stay in the ER. So you have a soluble protein of the ER. And also there are several components that are going to be on the endomembrane of the ER. So you have ER membrane protein, ER soluble protein. The same way, number two, if we just look at that, we have that Golgi right here. There is one here, and it's not shown, but just think about that. You have a protein right here, protein right here, protein right here, that you have Golgi soluble protein and also Golgi membrane protein. On this, a couple of membrane protein has been shown here. So it could be staying on the Golgi membrane for different functions, and it could be soluble proteins that are present in different stacks of the Golgi, because these are the resident enzymes of the Golgi that perform many modifications on the protein. So they are resident protein, resident ER, resident ER, resident Golgi. These are soluble and membrane. Then number three, once they pass all the protein, the final destination of the ER soluble protein is the ER lumen, ER membrane proteins in the ER membrane. Final destination of Golgi soluble protein is the Golgi stack, particular stacks, different ones. And then Golgi membrane is the final destination. Then the next one is lysosome. So the lysosome right here. So again, the lysosome will also have soluble proteins, which are inside enzymes, lysosome, soluble proteins, and also there will be plenty of proteins on the membrane of the lysosome. These are, so just think about this lysosome, all these soluble proteins are inside, and the proteins which are on the membrane, plenty of protein which are on the membrane, many different function on the lysosome. So both. Then number four, what you have, and the number five, what you have, they will be just only one of those categories. One is a membrane protein, and the plasma membrane is the destination. On the plasma membrane, you cannot have a soluble protein on the plasma membrane. That's just a common sense in the lipid bilayer. So you do not have anything final destination in here. It's only membrane protein that's going to the plasma membrane. And then we talked about the secreted. You cannot have any membrane protein that can be secreted. So here you have the secretion, right? Secretory protein. This is essentially the only type that is soluble. There is no membrane protein that could be secreted. So just these are the destination: soluble membrane, Golgi soluble membrane, lysosomal soluble membrane, plasma membrane, and secretory soluble proteins. That's it. So you can see that there are multiple destinations, 5 destinations overall, and 3 of them have membrane and soluble both, and the plasma membrane is secretory. So this is a huge pathway, and we call this pathway as a secretory pathway because all the proteins that are synthesized and secreted outside going to the plasma membrane travel through this pathway, all the way entirety for the, say, secretory protein or plasma proteins, or partially for the lysosome, Golgi, and the ER. And all of them will have essentially a targeting sequence. The targeting sequence is the ER signal sequence. So this is one of the most important part that we are going to talk about the next. So the first thing is that if you do not have an ER signal sequence or any signal sequence, it is ending up in the cytosol. When you have an ER signal sequence, the ribosome is going to be brought by supporting protein, chaperone proteins. We are going to see that it could be SRP or get complex protein onto the cytosolic side of the ER, where it will be translocated onto the lumen, into the lumen of the ER, or it could be inserted into the membrane. Now, the targeting sequence of the entire secreted pathway, so all these 5 different proteins, the targeting sequence is always the ER signal sequence. But what takes it to these different places that is not a ER signal sequence? That is called a retention signal, which is somewhere else in the protein other than the signal sequence, a retention signal. Very important. So ER signal sequence is where it is going to go to the secretory pathway. All of them are going to the secretory pathway. And the retention signal, the ER protein will have an ER retention signal, soluble, or membrane ER retention signal. The Golgi proteins will have a Golgi retention signal, Golgi retention signal, lysosomal retention signal, lysosomal retention signal, plasma membrane retention signal, or secretory proteins, which will be secreted outside. So remember that where the protein will go through the secretory pathway is a retentional signal. This is in addition, remember that this is an additional signal other than the targeting sequence, which is always ER for the entire secretory pathway for all these 5 destinations. 2 signals, ER signal sequence to make it a secretory protein, path through the protein, and retention signal, taking it or keeping it to a particular organelle in this left side only. Remember that we are not talking about the right side. So this is a very important concept. Remember that. And if you do not have any targeting sequence present in cytosolic proteins, it will end up in the cytosol. So we will come back to these targeting sequence, and we will also talk about the retention signal as we move through the material. So just remember to connect these dots. Now let's go to the next one. These are just some of the pictures of the ER, and these are really no questions from here. Just the rough endoplasmic reticulum are studied with the ribosomes, helping the protein synthesis, smooth ER, do many of the carbohydrate modifications and lipid metabolism. These are some of the structures, and it's a continuum. As I said, that there is no such thing that a separate rough ER, separate smooth ER. Some of the portions of the ER doesn't have the ribosome and appear smooth. Some of the portions on the side that you can see are studied with these ribosomes, which are actively synthesizing protein, either soluble or membrane proteins, and they are the rough portion of the ER. Now, how do you separate these things or make experimental component which can allow you to do experiments in this protein transport? So the first thing what you need to do is that you disrupt the cell by different methods like a sonication, blending. We have seen that, or passing it to a high pressure in a syringe, through a syringe, these cells after collecting. So all these endoplasmic reticulum, they immediately break up into small structures, which we called as a microsome. So think about that. Property of the lipid is such that if you just make a large glob of lipid, just think about that you have a beaker with water on top of that, you have an oil, and then you just close the top of the beaker and anything like that, or a screw cap, a tube, and you shake it. All these oil droplets actually separate out into tiny structures. And if you allow them again, then they will coalesce to form the layer again. So just think about that when you make microsomes or break up the cells, these tiny parts of them, the parts of the rough and the smooth endoplasmic reticulum, they break up into tiny structures. They do not leak the inside material. The integrity of the membrane is kept. The reason is that if there is any gap in between the membrane and the aqueous environment, automatically, immediately, all the phospholipids arrange themselves in such a way so that no material is actually leaked out. So there is no chance of the inside of the material to leak out. They just form tiny microsomal structure instead of these long structures of the ER. And we call them as a microsome. If they are studded with a ribosome, we call it a rough microsome, or it could be smooth ribosome. And this is the important part. Interior is biochemically equivalent to the ER lumen. And we can actually separate them by centrifugation. So these are a couple of pictures with that. So initially what you do, that you do the same, take out the fraction, mixture, homogenize, rough and smooth vesicles, and then you put them on a gradient. We have talked about that, how you can separate mitochondria, chloroplast, or the lysosome in the last unit 1. So you just load them on the gradient. What's going to happen, that the microsomes with ribosomes on top of them are heavier molecular weight wise than the smooth. They are going to go deeper in this area, whereas some of them will stay in here, and you just collect these fractions to essentially separate these rough microsomes, and you can do a lot of experiments with them. So let's take a look at what are the different types of experiment that you can do. So what we are seeing in this process, let's go to this process, that there was some evidence from late 1960s that the proteins that are synthesized through the ER, Golgi, going to the plasma membrane, secreted outside, must have a signal that takes these ribosomes and the mRNA onto the ER. But it was just not discovered until the signal hypothesis was put forward by Gunther Blobel, later won the Nobel Prize. And now we know it's no more a hypothesis, it's a proven fact that each protein does have a targeting sequence, which we call as a signal, targeting signal sequence or targeting signal for the ER. And this could be, here it is shown that it is at the very beginning of this, and it could be internal as well for membrane proteins. So we're going to see that. So what we are seeing in the picture is essentially a cross-section of the ER. This is the ER lumen. The dark part is the endomembrane or the ER membrane. And here is the lumen. Here you are seeing, in the first one, you are seeing some ribosomes, free ribosomal subunits, small and large. And here you are seeing the small and large ribosomes together with the mRNA 5' to 3'. And here you are seeing that it is secret or sitting on a channel, which is a blue channel-like structure. And we will know that this is basically known as a translocon, which transport a translated protein into the ER lumen or helps to insert it onto the ER membrane. All of the secretory proteins will use some type of translocation or translocon to translocate the protein. Now, again, here, there is one of the very important concepts that makes sure that you distinguish in between them. The translation is, so number one is a translation. So the translation is essentially reading of the mRNA and making of the protein. That is your translation. And the number 2 is the translocation, 2 different words, and make sure that you pay attention to these. Translocation is essentially the process of moving this protein either in through the translocon, either into the lumen, or later we are going to see, or into the membrane of the ER. That process, or the movement of the protein or insertion in the ER membrane or releasing into the lumen, is translocation. Translation and translocation. And we are going to see that there are 2 types of this. One is co-translational translocation, and another will be, so this is your A, and there is only one example that we are going to see in East, and another one is a post-translational translation. Post-translational. So just make sure that you understand the difference between the 2. A co-translational translocation means as the protein is synthesized, it is going into the lumen or onto the membrane. On the other hand, a post-translational translocation means that the protein is actually synthesized in the cytosol, the entire protein is synthesized in the cytosol and then brought to ER. We are going to see that by a signal sequence. This is simultaneously co-translational translocation, post-translational translocation. Protein is protein in cytosol and brought into the ER. So these 2 types we are going to see, and both of these needs a signal. Remember that. Not retention. We are not talking of the retention yet. This is a targeting signal so that it can be brought to the ER. So let's see that once this signal hypothesis was put forward, that they have also seen that there is a signal peptidase. So here you can see that that's in yellow. And this signal peptidase cleave the protein right here, or the signal. So the mature protein doesn't have that signal. It is removed and degraded. So the red portion is at the very beginning, signal peptide on growing polypeptide chain. It is cleaved by the signal peptidase, cleavage of this, and the mature protein doesn't have the signal, which is essentially degraded. This is just only one example of a protein, which is a soluble protein. Remember that it is going into the lumen. So the picture that you are seeing, that this protein, you know that this is a soluble protein. Remember that could be soluble. So where it could be the destination, depending upon that, this should also have a retention signal. It can be in the ER. It could be a resident protein of the Golgi. It could be going to the lumen of the lysosome, or could be secreted outside. It cannot be going into any of the membrane because it is a soluble protein, because it is in the ER lumen, which is aqueous soluble, not a membrane protein. So these are the things that you connect the dots for understanding the material. All right, now the signal hypothesis was put forward and how it was known. There is really a signal. Scientists did what is called an in vitro translation. To do an in vitro translation, it is essentially in the test tube in vitro, where you add the mRNA, you add the ribosomal subunit, and what they did, that into that fraction, they added, in one of them, they are without microsomes. In one of the other experimental part, they added microsome and then incubated that. And then they ran a gel to see that what are the outcomes of that. And how is this detail is not necessary. So how usually you will isolate these proteins? First of all, the proteins, when you have this process, you will have all these tRNAs, which is the transfer RNAs, you will have the mRNA, and you will have all the tRNAs, and there will be one of the amino acids that you have is essentially the radioactive, and the tRNA that will carry the methionine, because you know that methionine will be always at the AUG, the first codon, and there will be several in the protein. So all the proteins that are going to be synthesized is going to be radio-labeled. So you do the microsomes. And now what you do, this is a little bit of extra detail, that you do what is called an immunoprecipitation reaction. So precipitation. what is essentially doing that when you are in a test tube, you have the ribosome, you have the mRNA and this synthesized protein, all the microsomal protein, plenty of these protein. And now do something, say for an example, if you take all of these proteins and try to run on a gel and then stain it with a protein binding dye, there could be hundreds of these proteins will show up. You have no idea which protein is what. So if you radio label them, then only that radio label will be incorporated into the protein that is synthesized out of that. So this is the only protein that's synthesized. Everything is preformed. So no radioactivity will go in there. And on top of that, what you do, that you may have some resident proteins of the microsomes, and you can add some antibodies to that, which is added with a big agarose bead. So these antibodies, agarose bead. We talked about the fluorophore attached to antibodies. Here it is big agarose bead. So what happens that after you just do this, it will bind and it will just bring down all the microsomes, and it will just keep all other extra things that is not there outside the microsome. And then you run the gel. Now on the gel, you have to, after running the gel, you have to take an X-ray film and put next to it so that there is an autoradiograph developed. That means the protein that is radio labeled, this is radio labeled, that will have an impression on that X-ray film next to it. So what is happening that after you do this experiment, the fractions which doesn't have the microsomes and then ran the gel out of all those proteins without the microsomal fraction, and you will not, of course, not do any of those immunoprecipitation here, you are seeing a larger than normal protein size here. On the other hand, if you just take the microsomal fraction, essentially, they have seen that the molecular weight of the protein, which is radio labeled, is less. That proteins are negatively charged. So you load them at the top of SDS page. Just if you're not familiar with that, this is a prereq material. Just look it up, SDS page, and just take a look at that, what it is. And all these negatively charged protein move towards the plus electrode, which is at the bottom. And the molecular weights, which are heavier, are going to move through this amide like a sieve much slower. So here, the molecular weight is higher here. It's higher. And here it is lower. So here it is lower. So this is essentially the difference between those higher and lower. So what is happening in here is without the microsome, the molecular weight is larger because you have the entire protein right here and the signal sequence here. This is the entire protein that you are seeing right here. Whereas once the microsomes are added, the ribosomes are brought onto the microsomes and the proteins is extruded inside the microsome, and the signal peptidase cleave the signal. So you have only the blue part, which is without the signal. That's why the molecular weight is less. This is without signal, and because the signal is cleaved out without. And this was the first evidence that indeed the targeting to the microsome, which is the ER, is a signal based, and the signal is a stretch of an amino acid which is in the protein. With the microsomes, it can be targeted to the microsome. Without that, the signal is not deleted. So this was essentially the experiment that proved that how this signal hypothesis was asked. So this was the experimental part of it. All right. So after that, we are going into a protein, and here we are asking two questions. So the number one question that is asked is that cell-free protein synthesis, no microsome present, cell-free protein synthesis, microsomes present. So first of all, how do you know that number one, how you know that the proteins are really inside? The proteins are really inside. How do you know? We know that there is some signal involved, but how do you know that the proteins are not really sticking onto the outside? Because you immunoprecipitated these things doesn't give you enough evidence at this time that whether they are really inside or they're sticking outside the microsome. So this experiment tell you that how you can determine that they were really inside the microsome. So in the first one, that what they did, that they take the ribosome, tRNA, mRNA, everything, and they completed the protein synthesis, and they added the microsome after the translation. It allowed the translation, and they added the microsomes after that. The next one, what they did, that they added the microsomes at the same time when the translation is occurring at the same time. And it showed the co-translational transport and the removal of the signal sequence. And that is a separate experiment that we saw. Here, the idea is that how do we know that these proteins are really outside or they are inside? Now, what you have in here, that if you add protease to this experiment, number 1A, the proteins will be digested, degraded as they are outside in the reaction mixture. So you add protease to this and then run a gel, and you are not going to get any protein. So here, this is no microsome present. This is microsome present. So everything degraded. You just get some radioactivity at the very bottom. And these are all radioactively labeled. Just remember that. On the other hand, if you add protease to that, I'm reading that line by line, if you add protease to these experiment proteins, will be protected by the microsomal membrane as the proteins are inside. The proteases are protein, large hydrophilic molecule. They cannot pass through the membrane without any transporter, and there is none. And protease can't cross the hydrophobic membrane. So when you run the gel, you are going to get the molecular weight of your proper protein, which is just without the signal sequence, and it is well protected. Whereas here, the proteins are essentially not protected, completely get digested. So this is the experiment that shows that there is a translation happening with the translocation, which is co-translational. Translocation occur simultaneously. If you add the microsomes after the synthesis of the protein, it doesn't transport the protein inside the microsome. Only it will do that when you do the protein synthesis or the translation in presence of the microsome, so that they are happening co-translational transport. So this is the experimental evidence for that. So this is very important. You're going to expect some questions from those. So just make sure that you understand these couple of slides very, very well. What is shown that the signal peptide is cleaved, and this is the same material. So this material, and that's why I did not put anything in here. It is just another structure, just an individual protein has been shown. See the previous slide. Now, this experiment is just the continuum of this part. See that right here? You added proteases. You did not. You added proteases, protected, not protected. And think about that. We take now right here this. Now, okay, all right. So here, this is your A. Now we have a B. So our preliminary experiment tells you that the proteases of this experiment protected by the microsomal membrane, B. So we confirm that. Now, in a C experiment, we want to confirm that even more strongly. So C experiment. So from the C experiment is essentially, this is how you connect the slides, is this. So this is your C experiment. This is the confirmatory experiment that indeed the proteins were inside, which is just a continuum of that. So what we did here, the first part is just the B experiment of the previous slide. See that you have the microsomes, and they are the microsomes and the protein synthesis took place. They're all inside, and you take this essentially. Now you have two different fractions. One of them, this is your B experiment from the previous one. You added the protease, and there is no digestion of the secretory protein. And we have a C experiment right here, where what we did, that we took these microsomes and treated it with a detergent. See, the detergent will make holes these white parts are the holes in the membrane, because you know that the detergents actually solubilize the lipids and the proteins. And after treating the microsomes with a detergent, then you add the protease. And now the proteases will be able to travel inside the microsome through those holes that were made by the detergent, and the digestion of the protein will happen. So again, repeating your A experiment, proteins were outside, proteases added, they were digested. This was a previous slide. B experiment, we added protease, they were protected because we assume that they are inside, but just to make sure that they are really inside. So we treated another fraction of them with the detergent to make whole on those microsomes, then added the protein digesting proteases, and now the proteins were digested and they are not resistant anymore. Here they are resistant to protease digestion. Here they are not resistant to protease digestion once treated with the detergent. Pretty much it. So just make sure that you read this, because we are going to see that in some of the questions, that when you like experimental detail of this, like a hypothetical question, like if what's going to happen if we give you this scenario, you add the detergent, then you add the protease, what's going to happen? You have this experiment, you add the proteases, but you have not added detergent, what's going to happen? So this kind of variations of the question you can expect. All right. So after that, we are looking at some of the signals that what you have. The signals are protein delivery addresses. And these are hydrophobic, followed by some of the positively charged. You do not have to memorize any of them. This is just to give you a signal peptide, 35 amino acids. And remember that is deleted or cleaved from the mature protein. That's why they are losing that molecular weight, and that's why they are shorter. The matured proteins without the signal peptide are shorter than the proteins with the signal peptide, what is being shown. That larger than normal, this is the protein with the signal peptide. This is the correct size of the protein without the signal peptide, because that is usually cleaved, this signal. And here, we did not talk about that. See that this is your ER import signal, that's previous page. And here it is a retention signal. See that right here, you have an important thing that we talked about. All the proteins in the secretory pathway will have this signal sequence right here. Is the same in all of those 5 destinations: ER, Golgi, lysosome, plasma membrane, secreted outside. But if they are retained in the ER, you have a lysine aspartate glutamic, a lysine, we call this KDEL. KDEL in single letter word. This is your essentially the sequence that what you will have. Essentially, we are going to see that this is very, very important for your in some other places. You do not have to memorize that. Mitochondria, nucleus, peroxisome, see that all of them have some kind of signal resistance at that. And this is not needed, just gives you the idea. But just the idea of a retention signal. And the same way we did not talk about that, you will have a retention signal for the Golgi, you will have retention signal for the lysosomes, you have all those different signals for each one of the destinations. All right. So after that, we are talking about, we just saw that the ribosome, the translating ribosome, is brought onto the ER membrane, and the co-translational translation happens. But who brings it, how it is brought, and what is going on? We are going into the molecular mechanism of that. And here we need some detail. So for an example, the first thing that what we are seeing, that our ribosome will get into the small, large subunit, will read through the mRNA. And as soon as the signal peptide, here it is at the N terminal, NH3 is the N terminal, and this is a secretory proteins, for an example, which is synthesized through this pathway. As soon as the signal sequence comes out, remember that the signal sequence is hydrophobic. So particularly most part of it, this leucine, valine, isoleucine, phenylalanine, tryptophan, alanine, these are all hydrophobic amino acid. They do not like to interact with water. So they are thermodynamically unstable. And immediately upon the emergence, again, repeating the word, immediately upon the emergence from the ribosomal tunnel. So there is a tunnel right here in the ribosome through which the protein is pushed downward. This actually approximately 30 amino acid fit into that tunnel. So as soon as there are 30 amino acid put in there, this is going to come out of that. And this is like a super fast process. Now, there is a protein, a ribosomal complex, and this is a large protein ribosomal complex or ribonucleoprotein. You can tell that, you can say that, which is called the SRP, ribonucleoprotein, because it's made up of RNA and protein, RNA plus protein. So this is called the SRP. So the SRP does have some grooves which are hydrophobic. So immediately what it does, that with the hydrophobic groove, it will cover up the hydrophobic signal sequence. And we call this one as in the biological sciences, anywhere where something is hidden, we call this as sequestering particularly. So it is hiding that part. So it's hiding the hydrophobic sequence. And so number one, sequestering signal, this is number one. And number two is the SRP will arrest the translation. This is temporarily. So what is happening, that as the hydrophobic sequences interacting with these hydrophobic sequences, it is like a glue, it is stuck, and the ribosome is stuck in place. It's not going to ratchet the mRNA through that, and it will not push any protein for a little while. In this little while, this entire particle is bouncing around with Brownian motion, and the SRP will find an SRP receptor on the ER membrane. So here you have the ER lumen. This is the cytosolic side of the ER. And on the ER membrane, you have the SRP receptor, which does have an alpha subunit and a beta subunit. And there are plenty of them, many of them, because you have seen that all these studded ribosomes, they are all brought at the same time. Many protein syntheses are going on at the same time. So the SRP will find one of them, and it will bring the translating ribosome onto the ER membrane. And the SRP will bind to the SRP receptor. So this is the first step that is happening. So here, take home message that what is the most important part, or what question we ask here. So the question is that, look at that, the topology of the membrane. If we ask you that where the translating ribosome with the help of the SRP will bring a translating ribosome onto which surface of the ER membrane, ER lumen or the cytosol. Remember that it is bringing it in the cytosolic side of the ER, not the lumen of the side, because it is putting it on the outside of the ER membrane. So this is the most important part, is that sometimes we ask you that, say, which of the following is a property of SRP? So again, I'm repeating, property of SRP, very important, property of SRP. And I'm just talking about one at each time, that what you need to know. So number one, it targets the secretory polypeptides in the cytosolic translating ribosome, and brings the translating ribosome on the cytosolic face of the ER membrane, cytosolic face of the ER membrane, cytosolic face. That is the most important part, face. Remember that. Number two, it arrests translation. We already talked about that, arrest translation. And it binds to a terminal internal hydrophobic sequence. So remember that it binds to a terminal. Here it is a terminal, or we are going to see later that there could be a sequence in a protein where, if it is a membrane bound, the sequence could be internal for a membrane protein. Remember that? This is suppose a membrane protein. So you have a membrane protein like this. We are going to talk about that later. So it will sum up like right here. Here, this is the signal, SA signal anchor sequence. We're going to see that later. Signal. So the SRP may bind to a terminal, or it could bind to an internal sequence to bring that. But the most important part is that we can give you two options, that it will bring a translating ribosome on the luminal side of the ER membrane, which is essentially the exterior or the luminal side of the, you can say inner, or you can say luminal or exterior side of the membrane, that will be very wrong. It always bring it on the cytosolic face, which is outside the ER membrane, and the protein will go through a translocon. That's the machinery. So this is like a direct type of question that we are asking. Again, properties of the SRPs, this will help pretty much. All right, very important thing. So just remember and rewatch that what I said, 1, 2, 3, these are the properties. Very important. Next, some more properties. You can see some more questions out of that. Say, for an example, structure of SRP contains several proteins and RNA. Do not have to memorize these P54, 19, these numbers. These just denotes that these are different protein subunits, and these are the molecular weights. And you can see that there is an RNA with the secondary structure 5' to 3'. RNA is single-stranded, but it forms the secondary structure and looks like that they are double, but they are not. But different subunits interact with ribosome required for protein translocation. And this is what just only pointing out that we just talked about just before. These are some of the residues that bind to the signal sequences. See that right here? These are all hydrocarbon chains, and they are all hydrophobic. This is all hydrophobic. That is very important. And along with that, remember that it functions with the help of a GTP. GTP is also required. So several proteins, RNA and GTP. We have a separate slide, but I'm just adding in there. So remember that it's a ribonucleoprotein, and that's basically the structure. All right, so here structure, GTP is required for SRP function. So whenever you see that there's a star mark slide and there is something highlighted, you better know that that will be a kind of a question. So you absolutely need to know that highlighted part. GTP is required for the SRP to function. So remember that the GTPs, again, the structure detail is not necessary. If you just remember that, that's good enough. These are essentially the crystal structure of different homologs, where it shows that this is the GTPase domain of the SRP54 subunit. And this is a FTC homolog of SRP receptor and alpha subunit, which is essentially, this is the P54, you can see. And this contains that GTP binding sites, which is being shown in a molecular model. And that detail is not necessary, just important that it is GTP functioning. So now from this picture right here, this was the essentially that how the proteins are coming out, the signals are coming out, SRP is catching it, bringing it to the receptor. And here we are talking about the translocation of a soluble secretory proteins into the ER lumen, the entire part, and we'll have another picture for that to reinforce as well. Now, very important, because remember that whenever we are talking about soluble secretory protein, remember that destination could be, it could be retained in the ER, it could be retained or go to the Golgi, go to the lysosome, or it could be secreted outside. So these are the 4 fate or destination of these ER luminal protein. Remember that here it is temporarily, and it will stay if it is an ER protein, but this will be transported after modification and folding from the ER to the different destination. So let's start with that. So we already saw the SRP is binding to the SRP receptor, and we're going to see that there are 2 GTPs right here. There are 2 GTPs bound here, and these 2 GTPs on the SRP is going to be hydrolyzed once the SRP interact with the SRP receptor. And at that moment, the ribosome is going to be translocated onto a protein channel, which is called a translocon. This is like a very large protein. And think about that this is like a paper towel roll through the ER membrane. And through this paper towel roll, there is a gate. Gate opens once ribosomes sit on it. And through this paper towel's central channel, the protein is extruded into the lumen of the ER. And here you can see the signal, which is not shown. The signal is going to be cleaved by the signal peptidase. And this is your co-translational translocation. And here you have a luminal protein. This is in the ER lumen. So this is just an experiment. Detail of this is not necessary. That how it was really found protein is going through the translocon. So what the scientist did, you can just go to the book and read that, but I'm just in short, just an overview of that. They made an artificial mRNA from prolactin, which didn't have a stop codon. So what happens that if you do not have a stop codon, the tRNA remains, the last tRNA or the last amino acid it is bringing remains attached to the mRNA, and this entire structure is intact. It's until the mRNA, the ribosome reaches the stop codon, it's just not going to let the subunit and the tRNA and the other structures go. So this is stuck. And intelligently, what it was predicted that if you just can fit about 30 amino acids in the channel, they intelligently chose a residue called a lysine and modified that lysine residue, which can be activated by light. And upon activation, there is a cross-linking agent that can covalently bind it to this translocon, which is the Sec61 alpha. So this is a translocon from the East. Nothing but just a specific name rather than a generic name. So what they did, they allowed this translocation and the translocation essentially to happen. And what will happen that it will be stuck in this position after some time, because this once the tRNA reaches the very end of it, no stop codon, it is not going anywhere, but it will arrest the translation, and the protein should hang like this through the translocon. And they shined a laser light onto this. So this light activated cross-linking agent is activated, and it will covalently bind this protein onto the Sec61 alpha. Now what they did, this is the similar way that they disrupt all these microsomal structure, and then they did an immunoprecipitation reaction. And again, remember that there is always radioactively labeled proteins. So they used these agarose bead, and these antibodies are against the Sec61 alpha translocon. So essentially, it will bring down these antibodies. It will bring down, when you gently centrifuge them due to these agarose bead, this translocon. So what you should see in the translocon, that when you have this experiment, this is without the translocon, say for an example, and this is with the translocon. You are going to see this protein essentially right here. And what you are going to see, suppose you have a normal protein. A normal protein's molecular weight is much, much less, whereas the protein which is added with the translocon, it is showing a much higher molecular weight. The reason, it is the molecular weight of the translocon, because you have added the cross-linked it with the translocon. It is the molecular weight of the translocon plus the protein. If you do not cross-link them, this is the molecular weight of the protein only. If you just allow them to pass, and then you allow the translocon, you purify the protein and you run the gel, you're going to see the molecular weight of that. But you will see that drastically the molecular weight of the protein will change, because this is the molecular weight of this huge paper towel roll and the protein together. Here is the protein only, here translocon plus protein. So they were sure that this were definitely going through the channel of the translocon to the membrane. So that's the mechanism that how they found it out. And this is how it was discovered. All right. So here we are looking at the structure of the Sec61 complex, which is the translocon. So what we are looking at two different views. This is a side view, and we are looking at the paper towel roll from the side right here. This is the side. This is the pore through which the protein goes in. So if I just change the color, just right here, the protein goes in here through here like this. On the other hand, what you are seeing in the other picture, this is a top view. So I am looking from the top, and this is the paper towel roll's central channel. And this is the paper towel roll that we are seeing. And the protein is essentially going through here. So this is the protein. This is the top view, and this is the pore. Why this is important is, and there is a side exit through which the proteins that are going to go into the membrane actually exit the translocon. We are going to see that. So if we just redraw this picture to make it correct, that there is an idea that, think about that there it is like a clamp. you have a structure like this, and think about that there are certain type of proteins, or these are some of the residues that what you have. You see that right here in purple color, there are 2 alpha helices in there. And those purple color alpha helices, I can just make them purple probably. That will make sense right here. These are the alpha helices, which are the gates. So what happens that these gates actually move out. If we just think about that when the protein needs to exit from the sideways, they will move out and the protein will extrude into the membrane like this. So think about that. If we just talking about this, the protein will move through here, and now it is a membrane protein. This is the side view. This is from the top view. So this can open and close, lateral exit to the lipid bilayer. So this is the concept that what you are seeing in a translocon, and this is important for your membrane. The protein which is soluble doesn't need to exit. The entire thing will fall through the pore of that central channel into the lumen. But this concept is important to see the membrane proteins. So let's review one more time the translocation of soluble secretory proteins into the ER lumen that we already talked about is the same thing. And here you can see that SRP is bringing those. And remember that there are 2 subunits of the GTP. So just remember that picture sometimes shows you an empty space like that and occupied by the GTP. But remember that the SRP receptor, this is the important part that you can take a note of that. The SRP receptor, this is a GTPase. It doesn't contain or bind to any other thing. SRP activates the GTPase activity of the SRP, but the receptor itself is not a GTPase. It is not a GTPase. The GTP is bound to the SRP always. The SRP receptor only activates it. And now once the GTP is hydrolyzed to the GDP and the PI, see that right here, it just looks like that there is an SRP, which is SRP receptor is going that. But remember that both of these GTPs are the part of the SRP. And how we know this picture of the structure, there are these 2 GTPs, and these 2 GTPs are sitting on the SRP receptor, and each will be hydrolyzed to the GDP to PI. And this structure essentially going to get separated from the SRP receptor. And once this GTP is hydrolyzed, the ribosome along with the mRNA and the part of the protein is going to be transferred onto the open translocon. So this is needed. So just remember that's how the sequence of events is happening. The SRP with the GTPs bringing the ribosome onto the SRP receptor. SRP receptor activates the GTPase activity of the SRP itself. So SRP is hydrolyzing the GTP to the PI. In that condition, the SRP receptor is separating from the translocon and transferring the ribosome and the mRNA on the translocon, and the gate of the translocon opens. And now the protein synthesis starts. Remember that as long as the SRP is bound to the ribosome, there was a temporary arrest of translation. Once it sits on the open translocon, translation resumes, and translocation also happens at the same time. There is nearby signal peptidases, which are membrane-bound enzymes that will cleave the signal sequence, and this will be degraded, and the entire protein will fall into the lumen once it is done. Once the protein goes and the ribosome separates out, the translocon will close. The ribosome and the translocon interaction opens the gate. When the ribosomes are not interacting with the translocon, the gates are closed. So this is, again, soluble secretory protein. This could be ER protein, Golgi protein, a lysosomal protein, or proteins that can be secreted. So this is just only your type A. Now, this is just only one example. Remember that we talked about this, and we're going to talk about another one. We also see the post-translational translocation in the yeast, where that entire protein was synthesized in the cytosol, and then this signal sequence, this is a different type of translocon, Sec63, there is SRP involved, which is not shown in here, that binds to this and brings only the protein, which brings in the protein, and then the Sec63 complex delivers these BIP or the BIP proteins, which are like bead-like structure onto the protein. The reason is that when you have a post-translational translocation, not co-translational translocation, see that during the co-translational translocation, the ribosome is sitting on top of this, so the protein cannot slide back. But here, when the only protein is brought onto the Translocon, this can go back and forth. This is being shown by the two arrows. Just make sure that there are tiny details in the pictures. These two arrows means that the protein can backslide and get out without getting in there. So how the backslide is prevented? As soon as the protein is inserted to some extent into the lumen, they are added with this BIP protein, binding protein, BIP, and they are just like beads. The BIP will not allow the protein to backslide because they are much larger than the lumen of the Translocon. So they will stay in the lumen, and once they are folded, mature proteins, mature soluble secretory protein, once they are folded, the folded protein has a much larger diameter and it's 3D, so it will not slide back either. So only the protein in its linear unfolded form can go back and forth slide. And this is a example of a post-translational translocation that is seen in the yeast sometimes. All right. So after that, let's take a look at the insertion of membrane protein. So this is all about how a secretory soluble protein is extruded into the ER lumen. The next part, membrane proteins are inserted into the ER membrane. And again, remember that when we were talking about that, this could be resident ER membrane proteins, Golgi membrane proteins, lysosomal membrane proteins, or plasma membrane proteins. They are not soluble, but they will have a hydrophobic sequence, which is inside the hydrophobic part of the membrane. All right. So here, just a concept, no specific question, essentially, topology. So what we mean by the topology, that if you just look at the cytosol, this is the cytosolic side of an ER membrane. This is the luminal or the exterior side of the membrane. Now, you can have a long N-terminal on the luminal side. You have a short C-terminal on the cytosolic side. On the other hand, the opposite, N-terminal short, C-terminal long. N-terminal long, C-terminal short, long C-terminal. So if you have a single pass protein, doesn't matter, the protein can have all different combinations of the topology. The N or C terminal could be either side, cytosol or luminal side. Either side could be big or small. And these are a couple of examples. Do not have to memorize any of those names specific to that. On the other hand, you have another type, which is the multiple pass proteins. So the multiple pass proteins, essentially, that we were seeing, this is GLUT1. Just only one thing, just you have to remember that if you have an even number of multiple pass proteins. So if you just look at the GLUT1, it will be 12, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So even there are 12 numbers. There are many, many proteins. If it is an even number, then the N and the C, they will be on the same side. Remember that? If there are some proteins which are even number, the N and C terminal could be on this side, N terminal on this side, C terminal could be on this side as well. But the bottom line, if you have even number of transmembranes, the N and C terminal end up in the same side, either cytosolic or exterior. If you have odd numbers, they are on the opposite side. So here, see that N terminal is in the luminal side, C terminal in the cytosolic side. On the other hand, it could be the other way as well. C terminal could be on the luminal side, N terminal could be on the cytosolic side. You have that kind of examples as well. So it can like you can have, I'm just showing is that it could be just going back and forth in each directions, no matter what. So all permutation and combinations are possible, but take home, even number, the N and C terminal are on the same side, odd numbers, N and C terminal on the opposite faces of the membrane. So that is the most important part. All right. So one thing that is actually important in here, that if you just think about that, we have any protein, and suppose in the ER, we say that N-terminal is on the lumen or exterior. Suppose this protein is becoming a plasma membrane protein. Suppose this protein grows to the plasma membrane. And we ask you the question, very simple, but sometimes when you connect the dot, it just becomes complicated. If this becomes a plasma membrane protein, where the N-terminal will be? The N-terminal will be on the exterior, because see that the N-terminal in the ER is on the luminal side. That is equivalent to the exterior. So if this protein becomes a plasma membrane protein, it will be on the exterior side. See, the C-terminal is on the cytosol, so it will be on the cytosol as well. So if I just take this protein and draw that a picture that where this will be in the plasma membrane. Suppose this is plasma membrane. This is trafficked through the Golgi, loaded onto the vesicles, then go to the plasma membrane. You are going to see that this part right here, I'm just in short, N-terminal is on the outside right here. And then you have the transmembrane part, and then you have the C-terminal. This is in the cytosol. See, the C-terminal is in the cytosol, N-terminal is in the outside or the exterior. So just make sure that when we ask you a question with a picture, say, for example, we give you a picture of an ER protein. This is how it is inserted into the ER, how it will be in the Golgi. Same way in the Golgi, it will this portion will be on the luminal side. This will be on the cytosolic side. If it goes to the plasma membrane, this will be on the exterior. This will be on the interior. So the topology never changes. It carries through wherever it goes in the same fashion. So that is your take-home message from the topology overall. All right. So after that, what we are looking at is the membrane proteins and single pass, multiple pass. And they are different types. And you do not have to type 1, 2, 3, 4. And you just have to kind of like an overview of that. We are not going to ask you which is in type 1. Where is your N-terminal? Where is your C-terminal? In type 2, where is your N-terminal? Where is your C-terminal? We are not going to ask you that. So do not have to memorize that. Just the mechanism for each one of them, that how they are done. That's number one. And then number two is the topology. On the other hand, you have all these example of these. You do not have to memorize any of those example either. So that is the important one. So just need to know some of the basic facts for each one of them and the topology. That is the most important part. So let's take a look at that. One thing that is important in here that the name actually suggests. So other than that, I'm just very specifically picking up that one thing to remember, that you see that right here, type 1 C-terminal on the cytosolic side, N-terminal, difficult to remember. You have to memorize. Type 2 C-terminal here, N-terminal here. So it could be a little bit confusing and it could be difficult. But if the name suggests it, you should be able to memorize that. And that's why I'm only pointing out this. The tail-anchored protein. See, the tail-anchored protein means the tail is in the membrane. So essentially, remember that the tail-anchored protein will have a hydrophobic sequence where the tail is anchored. And if the tail is anchored into the membrane, the hydrophobic sequence, this is your hydrophobic sequence that goes into the membrane, must be at the C-terminal. So this is a concept, not a memorization. Remember that. Here they are internal. But in this one, if I say that where it is, you do not have to memorize that. Just the idea is that it's a tail anchor. That means the tail is inserted into the membrane. And in a membrane, only a hydrophobic sequence can go in there. So the hydrophobic sequence must be on the tail. So hydrophobic. So I'm just specifically telling you this to take home message from here. C-terminal, hydrophobic sequence. All right, so you have all the different types, and let's see that how they are synthesized one at a time. If you get the hang of it, you will be able to predict for each one of them that how they are synthesized. It's no big deal. So let's take a look at the type 1. So the type 1, just remember that, is very similar to your soluble protein, which does have an N-terminal signal sequence. And now what we will also introduce along with the signal sequence, we have a signal. We will see that there will be another thing that will come out. Number two, we will see hydrophobic sequences, hydrophobic internal sequences. Hydrophobic, I'm not writing signal or anything, hydrophobic internal sequences. And these hydrophobic internal sequences could be of two types. You can have an SA, which we call as a signal anchor. Signal anchor. And then you have a B, we call STA, which stands for stop transfer anchor. Transfer anchor. Remember that the SA is still a signal that binds to the SRP, responsible for that part of the protein and the ribosome to brought in there. STA is an internal hydrophobic sequence, but it is not a signal. It just temporarily arrests translation to help the translocon move that part into the membrane, not a signal. So there is a difference between SA and STA. We're going to talk about that. So let's take a look at what is happening here. So first, this protein, when it's being synthesized, remember that N-terminal was associated right here, which is now cleaved by this, and the protein is now extruded. So for this one, you already have the signal at the N-terminal. This is your signal sequence. Now, protein is extruding into, and then you have the next hydrophobic sequence came out. Next hydrophobic sequence came out of the protein. This is the sequence which will be inserted into the membrane. But this is not a signal because the signal was already N-terminal and the protein on the SRP brought the ribosome on the translocon upon receiving this signal. And the ribosome is sitting right there. This is not a signal. This is a stop transfer anchor or STA. What it does that it interacts with the hydrophobic sequences of the side channels. And if you remember that we just went a couple of slides back right here, that I said that once these hydrophobic sequences interact with these two purple things, it opens this lateral exit and it takes a little time for the proteins to laterally diffuse out into the membrane. And that is the reason that for that purpose, it stopped the translation temporarily. So that's all it is. So if we just go back right here, the stop transfer anchor sequence momentarily stop the transfer and the translation, and the protein is diffusing through that lateral exit in that paper towel roll into the membrane. Once it is into the membrane, this is a hydrophobic sequence, the rest part of the protein is synthesized. Now you can see your N-terminal is in the luminal side. You have a hydrophobic sequence, and the C-terminal is on the cytosol. So this is how this type 1 membrane protein is inserted into the membrane. Only one signal sequence followed by a stop transfer anchor sequence. All right, so let's go on to the next type. So this one is a type 2. The N-terminal is in the cytosol, so the C-terminal is in the lumen of the ER. So here there is a little bit problem because you have to lower the protein like an U-shaped structure. Like think about that you just holding a pipe, and then you are just taking a string and you are holding the both end of the string and gradually lowering both the strings through the channel into the lumen. So that's basically how it is. Now, a couple of different important things here. So these are the ones that is important rather than where is N-terminal, where is C-terminal. So from N-terminal all the way up to this point, this blue part, see that there is no hydrophobic sequence, there is no signal sequence. So this part essentially is synthesized in the cytosol by the ribosomes hanging in the cytosol because there is no signal. There is no signal. Now what happens that you have an internal signals. Remember that it's an hydrophobic. It's an internal signal comes out, which is this. only at that time the SRP will bind to the ribosome, arrest the translation same way, and bring this entire structure onto the translocon only upon appearance of this internal signal anchor sequence. Why it's a signal anchor? Because this is the signal, and this is going to anchor the hydrophobic sequence into the membrane. That's why it's a signal anchor. Last times, just if you compare that, last time, the first signal sequence brought the ribosomes onto the translocon. So second one was not a signal, it was a stop transfer for transferring that into the membrane. In here, this is the signal for bringing it to the translocon. So now what happens, that this is the important part that pointing out, you are going to see that, and this is very important. There are some plus charges on this part of the protein, plus charges. Now, the mouth of the translocon, you have plus charges on the mouth of these translocon, plus charges right here. So the plus actually repels. So what happens, that this part, which is the blue part that should remain in the cytosol, due to this plus charges, will never be inserted or lured into the lumen of the translocon. So the first part that will go inside the lumen of the translocon is essentially the signal anchor sequence. It will stop transfer for a little while, stop translation, as you can see, and it will allow it to diffuse into the membrane, while the rest part of it doesn't have any plus sequences. So this will come out and drop straight through this channel into the lumen. Again, repeating that, what are the important things in here? Number one, first part with these plus sequences synthesized in the cytosol, number one. Number two, the signal anchor sequence bind to the SRP, and only at that time the ribosome is brought onto the translocon. The signal anchor sequence bringing SRP. So you will remember that, SRP. And number three is the arrest that will help the hydrophobic part to move in through, and then resume. Resume means that translocation will resume. And only remember that the plus charges doesn't allow this cytosolic part to fall through in here due to repelling plus residues on the mouth of the translocon. So this is your type 2. Once you get the type 1 and the type 2, then if just two examples of this, then the rest will become easy. So let's go on to the next one. See right here, this is the type 3, where you have a short N terminal, long C terminal, but exactly like the type 1, except remember that, except the N terminal signal sequence. So here there is only one sequence. There is only one sequence, and that is your signal. So this signal is SA. This is a signal anchor sequence. See, the type 1 already had the signal in here, which is absent here. So this is the only sequence. So this is your signal anchor sequence. So what is happening? This will be this part in terminal is going to be synthesized in the cytosol. When this part comes in, we think that there is an SRP involved, just not very clearly known, but some chaperone protein, if not exactly SRP, that will bind to this signal anchor sequence, bring it to the translocon, arrest the transfer and the translation so that it can diffuse through here into the membrane. And then immediately after following the hydrophobic sequence, you have some plus charges right here. And the plus charges will not allow this part to go through the lumen, and this part will remain in the cytosol. So that's it. So this is exactly type 1, except the N-terminal sequence. And you have only one hydrophobic sequence, which is the membrane signal, membrane retention signal, because it's hydrophobic, but it's a signal anchor because it is the only signal. All right. So after that is an important protein, and definitely remember this one very, very well, because there is a difference in here. First of all, it's a tail-anchored protein, so the hydrophobic part is on the tail. Remember that hydrophobic tail, or hydrophobic C-terminal, which is essentially the tail of the protein. all the other examples that we saw, and even what we will see later in the multipass, we saw so far, except one of the exception in the East, we saw all of the translocations are co-translational. Co-translational. Right here. Co-translational. Except this one, this is not co-translational, but this is post-translational translocation. Remember that I introduced that word, that the post-translational means the protein is entirely synthesized in the cytosol, and only the protein is brought onto the translocon, not the ribosome. Ribosome synthesizes the entire protein in the cytosol. So that's why the translation happens. Translocation happen post-translation, not simultaneously. So this is an exception. Why this is so? The reason is that the hydrophobic sequence is at the C-terminal tail. So unless the protein is completely released from the ribosome, the C-terminal end cannot come out of the ribosome. And unless it is released, there is no signal. That is the only signal. So the ribosome has to release the entire protein, and it is only possible if it is doing in the cytosol. So once the protein is released, this is hydrophobic, thermodynamically unstable. Immediately, a couple of protein, GET4, 5, and SGT2. We call this as a GET complex. Do not have to memorize individual names. Just remember, this is we call as a GET complex protein. They have the hydrophobic sequences, and they immediately come over, cover that. So these proteins cover or sequester the protein first, and then hand them over to a GET3 complex. So just remember that. This is equivalent to SRP, but SRP brings the ribosomes. This brings only the protein. This is one of the difference. SRP works with a GTP. SRP works with GTP. This works with ATP. So these are the similarities and the differences. So know the differences and the similarities with the GET3. GET3 only gets the protein to the translocon, works with the ATP hydrolysis. SRP brings the ribosome and the translating ribosome onto the translocon, works with the GTP hydrolysis. So these are important consideration from this. So the GET3 complex, again, cover this one up and bring it to the GET1, GET2 complex, which is the translocon, and hands over the protein upon the ATP hydrolysis. And then the C-terminal is inserted into the membrane. Now, listen to this very, very carefully. This is a high chance of people missing this question. I'm putting it like a 3-star. So if we ask you that which of the following complex can bring a post-translational translocating protein, translocation, can do the post-translocation, translational translocation, which can do in absence of ATP. This is one of the version. Absence of ATP. Post-translational translation in absence of ATP. This works with an ATP, so your answer is none. So post-translational, if I just say that it's absence of that, none. This is your question one variation of the question. Which complex can bring a post-translation or do post-translational translocation, or can bring a post-translational translocation in absence of ATP? None, because GTP is co-translational translocation. The second one gets everyone. So remember that. The second one is more tricky. So I'm just going to draw that. Second one, I'm just changing the word from here. Again, which complex can bring or do the co-translational translocation. Remember that, translocation. Which complex can do post-translocation in absence of GTP? As soon as you see that absence of GTP, people jump onto the GET3 complex because it works with an ATP. What they miss is co-translational. In absence of GTP, Yes, GET3 works with the ATP, but it does not do co-translational translocation. It does post-translational. So again, if it is co-translational, your answer will be again none. So just make sure that what you do. So the take-home message, that without confusing with anything, your SRP is co-translational. GET3 post-translational. Co and GTP, and post and ATP. If you just remember these mnemonic, you will not remember. SRP, co-translational translocation, GTP, GET3 complex, post-translational translocation, ATP. If you change this co to this and co to this, there will be wrong answers, or none of the answer will not be correct. So just make sure that you spend time with these. These are some of the in-between line questions. So just make sure that you get this one and make your notes correctly on this one. All right. So after that, we are looking at insertion of the multipass proteins, and it is a repeating process. And let's take a look at that once you have the internal sequence, first one is an SA sequence. That is your internal signal, which is the signal anchor sequence. So first one is the signal. Now what happens that once it is inside the translocon, you have the loop. The second one is not a signal because already the ribosome is on the translocons with the signal. So this is stop transfer. Now the ribosome gets dissociated and hanging in the cytosol. So once the third sequence comes out, this needs to be inserted into the translocon. So the third one is again a signal anchor. Remember that. This red one is again signal anchor. Then after the loop, you have the stop transfer anchor. Then the ribosome gets separated. And then for the third one, or the third set, the first one, red one, you still needs to bring that one onto the translocon. So this will be again an SA or signal anchor followed by the stop transfer. So here, see what is happening, that if you have an SA and STA, they are alternating. SA, STA, SA, STA, SA, STA. So this is how the sequences are. And we are going to see that there are some variations for that in a second. So here the protein is synthesized just like your type 1, where the N-terminal signal sequence not shown, which is essentially right here, that was cleaved, that is not shown. Once the protein is synthesized, it is cleaved from here and transferred by an enzyme called GPI transamidase, not aminase. So if you see the transaminase with an N as a destructor, do not choose this. This is the correct answer. So these are very specific, and this is transferred onto a GPI anchor. So what is the advantage of that? This is a glycophosphatidyl'inositol anchor, which is a lipid, and these have much more mobility compared to the protein inserted into the membrane, and they have high affinity for the lipid rafts. So this part is degraded and this becomes a lipid anchored protein. So the protein is first preformed, inserted into the membrane, then by the GPI transamidase transferred onto the preformed protein anchor. So that's important. All right. So after that, what we are giving you is that we have these type 1, 2, 3, 4, 5. These all have a linear sequence, and practice that how you can draw these in the other ways in the membrane. So if I just draw this, I will draw this protein like this. You have the blue part right here, blue part right here. So this is the signal sequence. So this is your N-terminal. This is your signal right here. This is your STA, stop transfer. This is your STA because this is already a signal. And this is your C-terminal. And this is your cytosol. So if I have to draw this protein on the membrane, I will draw it like this. So practice for all of them, that you just have this membrane in here, you have this in here, and the cytosolic side will have the plus signs. So if you see any plus signs, that will be always on the cytosolic side. And this is an only signal. So that's why this is not an STA. This will be an SA. You can see the cytosolic side has the N-terminal right here. And this is your C-terminal. This is your type 2. And see that you have the cytosolic sequence right here on the C-terminal. So this is how you practice for each one of them. You will see a critical thinking question similar to this. So practice those for each one of the types. The next one that what we are looking at, that type 4A. So here you have an even number 2, 3, 4, even number of transmembrane domains, and see that what is happening, that here N-terminal is on the cytosol, so you have the plus. So even if you just do not write plus or minus, remember that if you see a plus sign, that part of the protein is going to be on the cytosol. And the first sequence is SA, followed by an STA, SA, followed by an STA. This will be for the even number. If you, on the other hand, have an odd number, remember that odd number, the N-terminal is on the one side, lumen, and the C-terminal is on the opposite side, you may have one extra SA sequence at the bottom. So here what is happening, the first SA sequence brings this protein. So why you have an extra sequence here? This protein is, suppose this protein is synthesized, no signal here. And then you have this right here. And you have, this was the first signal that the SRP brought in here. But after inserting this portion is synthesized in the cytosol. And now the next signal comes out. Someone has to get this signal to the membrane protein again. So this one is also an SA. So this one will, again, if I just redraw this back again. So this will be right here. I'm drawing the type 4B. You have the second SA sequence, SA, and now it will be the same. Now it is inserted into the membrane. You have the loop right here. Right here, you have the loop, and then followed by the red right here. So you will see that the SA and STA will follow. And now you see that the cytosolic side has both the plus ended. So where are the plus charges? Where I will put the plus charges? Remember that I will put the plus charges right here on the cytosolic side. This is the plus charges on this side. So this is the cytosolic side. This is the luminal side or exterior side. And this, so you have an SA followed by an SA. And now after that one, what will happen? SA, STA will SA, STA, SA, STA will follow STA, SA, STA, SA, STA. See that it is exactly the same for the even number, just only at the beginning, because it's just one extra loop. You have an extra SA sequence at the very front. All right. So after that, these are called the hydropathy or hydrophobicity plot. What you do, that any number above the 0 line, when you see peak, that is an hydrophobic sequence. So it is a stretch of 20 amino acids are taken and computed out that their hydrophobicity or hydropathy profile, how hydrophobic are the 20 stretch of amino acid. And looking at that, you can tell that where are the transmembrane domain of a protein, just looking at its sequence. From the sequence, you can tell. So typical question that we could give you the scale. So this is your 0 scale. So right here in the middle, it will be 0. Top, it will just go 1, 2, 3, 4. So up means more hydrophobic. Remember that, more hydrophobic. The bottom means they are hydrophilic. So what you do, that we can just give you something like this. You just only count the number 1, 2, and 3. How, if I just give you a graph like this and ask you that how many hydrophobic sequence this protein does have, anything other than above the 0 line, you have 1, 2, and 3. So you have 3 hydrophobic sequence. And remember that, see that here you have these tiny ones which are omitted because they are not really very steep. I will not do any of those. Just above the 0 line, they will be clear cut, no confusion. Just count the number above the 0 line. That will be your number of hydrophobic sequences in the protein, in a transmembrane protein. That's basically very straightforward. All right. So the membrane transport, and we are going to look at that all these ER resident proteins play a role in a glycosylation, disulfide bridge formation, folding newly synthesized secretory proteins, and what they do. So here we were talking about these resident proteins, and these are all your ER resident proteins, different ones. They do different functions, and they are soluble proteins, and they have a retention signal. We're going to talk about that a little bit later, but they are all proteins that stay in the endoplasmic reticulum. So from here, you can see for each one of them, we really do not go into the detail of the mechanism. but just for each one of them, know that what their function is. Say, for example, the binding protein BIP, if you just go back all the way up and just one line, what it does, it just prevents the backsliding of the protein. So that's basically just one line function of that BIP. You have the peptidyl prolyl transferase. You can see that this one helps in the rotation about the peptide bond and helps in protein folding. So that's just overview of the protein folding. Mechanism not required. The next one, protein disulfide isomerase or PDI. Just remember that it just helps to form or reform the disulfide bridges and help in protein. So again, protein PDI, protein disulfide isomerase, do not have to go into any of the mechanisms of these, but just this is just it's what is showing is the disulfide bridges were formed in different parts, and it's rearranging these with correct disulfide bonds. So it can form and reform disulfide bridges and help in proper refolding. Just one function. So here, it just shows that all the proteins together. See the BIP help in backsliding and also other things. And then you have a PDI for helping in the disulfide bond formation. And then you have these dolicol phosphate, which brings all the oligosaccharides that are added onto the protein. So all these reds and the blues, these are N-acetyl glucosamine and the mannose that are put onto that protein for proper folding. And here you are seeing calnexin. This is also one of the quality control protein. Calreticulin is also another quality control protein. They have a binding groove for the glycosylated proteins. And by binding at the glycosylated protein, they can figure out that whether the folding of the protein is going correctly or not. So calreticulin, calnexin, these are quality control proteins, overview of that. And here it's just shown that this is HA protein, which is the hemagglutinin for the influenza virus. Remember that the virus hijacks the cell's machinery, and the spike proteins on the spike protein trimers on the influenza virus. When we talk about H1, H5N1, the H's are those, and this just shows that how they are folded properly. Again, no mechanism is necessary, and none of these does have any star marks. So directly, you do not expect any problem with that. Now, remember that here, this is important. Unfolded protein response, and in short, this is known as UPR. So this is a big thing in the cell biology. Once protein is not properly folded, how you degrade that protein. So the first thing that is done, that you have these unfolded proteins, and there are some IRE monomers. These are ER resident membrane proteins. So now you are see that all these proteins are ER resident soluble proteins. And here you have an example of IRE1, which is an ER resident membrane protein, and they are bound with this BIP binding protein. So when the proteins are unfolded, they are bound to the BIP. And once the BIP actually leaves these IREs, the IREs dimerize to form an endonuclease. So monomer to dimer. Now they are functional. So what they do, that they do have a part of them sticking out into the cytosol, and they actually splice this HAC1 mRNA. Now, these are the reason that we need a little bit of the previous idea about how the cell actually works. So here you have the nucleus. So I'm just trying to draw with a little bit of pore in, say, for an example. So these mRNAs were formed in here, and they do have a middle stretch. I'm just for middle stretch right here, middle stretch, and they're formed in here, and then they actually come out from the nucleus. So this is your nucleus. They come out like this, and they do have a space in between right here, which is what we called as. So this is an mRNA. So now, when an mRNA is formed in the nucleus, they have sequences in between the different functional part of the mRNAs, which are called introns. So just you can Google these words out, and this blue part right here is an intron. And the this is, think about that this introns are the junk sequences. Because when a gene is transcribed, they are not next to each other, but they are cut out in different spaces so that they are not lost if there is a chromosome breakage and those things. So these are some of the measures for that. So this is intron. Now, the first part, this is the coding region of the mRNA, and we call this part as an exon. So here, this is your exon 1, because this is the first one. Then you have an intron. This will be our intron 1. There could be many of them. And then this will be your exon 2. Exon 2. So when the mRNAs are produced, they are produced with multiple exons. Here, just only one is shown, but it could be 3, 4, 5, 6 number as well. Multiple introns, multiple exons. So in the nucleus, remember that here, this is an exception. In the nucleus, all these introns are of the mRNA, and the mRNA with exons and the introns are called pre-mRNA. They are synthesized as pre-mRNA. They are spliced out. Once they are spliced out, the exons are joined together, and it forms mature mRNA. Mature or spliced mRNA. And most of the mRNAs are matured and spliced, and they are transported into the cytosol, where they can bind to the ribosome and translate it into the protein as required. The HAC1 mRNA is one of the exception that is transported from the nucleus in an unspliced format. So you have an exon right here. You can see that just this is a cartoon exon. This one is an intron, and this one is another exon. So you can see that the first one is exon 2. This can be exon 2, but I'm just like is exon 1, intron, exon 2. Remember that, again, repeating, most of the mRNAs are spliced and the exons are joined and transported as matured mRNAs ready for translation. Not this one. They come out of the nucleus as unspliced mRNAs. Now what happens that when the IRE1 is activated by the dimerization, they splice out the intron and you have the matured spliced mRNA. And this can be translated into the HAC1 transcription factor. So this is your transcription factor. So what this transcription factor is essentially doing. So this is just the process. So you have to just remember that you have an unfolded protein 1, 2, then you have a 3, 4, 5. So just know the steps. Just know the steps step by step, very straightforward. We give you all these, that unfolded protein, binding protein binds to the protein. IRE dimerizes. The HAC1 mRNA is spliced out. The spliced mRNA is translated into HAC1 transcription factor. Now, the next part, people don't follow through. Remember that what is going to happen, that you have this cell right here, and you have the nucleus with the double membrane. So just remember the nucleus right here. And the nucleus contain the DNA. So you contain the DNA right here. The transcription factors HAP1, they are produced in the cytosol. These are produced in the cytosol. They have a nuclear localization signal. They have a nuclear signal. So these transcription factor proteins are going to go all the way inside the nucleus. And if it's green, they will bind to certain portion of the DNA, and they are going to make the mRNAs for the protein degradation machinery. mRNAs for protein degradation. So this is half of the story. So remember that protein degradation. You do not have to go into that, but just interested that what are the protein degradation machinery? Remember that. Remember this word, that is a proteasome. A proteasome is a cylindrical structure. You can just Google it out or just wiki it just for one line. Proteasome is one of the protein degradation machinery. And how a protein is degraded, a protein is labeled by something called ubiquitin, UB. These are on your textbook, but that these are not necessary. UB. So the proteins are, when added with these ubiquitin, they are destined for degradation. So any of these proteins, they will be added with the ubiquitin by a protein called ubiquitin ligase. Ubiquitin ligase. So the ubiquitin ligase is going to add all those ubiquitins. They are going to now read by the proteasome to degrade all these proteins. So making of the proteins of the ubiquitin ligase and the proteasome, which are the protein degradation machinery, the synthesis of these proteins requires synthesis of their mRNAs, and this is triggered by the HAC1 transcription factor. So just remember that this is just only the very first step of that. The whole ball game is after that, making the mRNAs and the proteins to degrade the machinery. So this is just the process of thinking about that this is just the induction of the degradation machinery to be synthesized in the cell. This is your unfolded protein response. After that, we are looking into glycosyl transferases, and these are big thing in here. They glycosylate proteins. So we'll go into the detail of those. So the first thing what we are looking at is the N-linked glycosylation. Remember that N has nothing to do with the N-terminal. This could be anywhere. N is the single letter word for asparagine. So N is asparagine, then X, and if you have a serine or threonine, this is usually a sequence where the glycosylation takes place. Asparagine is N, that's why we call this N-linked, nothing to do with N. What are the glycosylations that is done in the ER? You have 2N-acetylglucosamine, then you have 1, 2, 3, 4, 5, 6, 7, 8, 9 mannose, and 3 glucose are initially put together. And then there will be a sequential trimming of this, and then they are ready to go to the Golgi in the next step. So here, what we are looking at looks a little intimidating, but all you have to remember from here is that this is a dolicol phosphate. Remember that this is a flipase. We talked about that in the unit 1. This is a flipase. What it does, that the dolicol phosphate plays a key role because it brings the glycan precursor synthesis for N-linked glycosylation. So see that on the dolicol phosphate, you have the first N-acetylglucosamine, second N-acetylglucosamine, and then you have sequentially all these mannose. And now it flips by its flipase activity and brings this entire structure here. Now, other dolicol phosphates are just essentially adding these glucose right here. And after that, all these glucose and the mannose and everything is completed together. So on top of this, see that it's bringing more mannose. All these mannoses are there. And then this is bringing in the glucose. These are all the glucose. So ultimately, the dolicol phosphate are done with 2N-acetylglucosamine, 9 mannose, and 3 glucose. This entire glycan or the glycan precursor is going to be transferred onto the N-linked glycosylation, and then it will be gradually trimmed. That's what it is. So take home message, dolicol phosphate is a flipase. It's a membrane lipid, dolicol phosphate, and it brings the glycan precursor for the N-linked glycosylation. Stepwise, nothing is required. All right, but the name dolicol phosphate is important. So here it is showing dolicol phosphate transfers the entire glycosylation onto the protein right here, and the dolicol phosphate will go back and do this function. Now, in the rough endoplasmic reticulum, we call this as a trimming of the glycan. Some of these residues or the sugars are taken off. So all the glucose in green is gone, and one of these mannose is gone. So it ends up with this. So we are going to see this. And again, these are the separate steps. You can see that once all these trimmings are done and you have 2N-acetylglucosamine and you have 1, 2, 3, 4, 5, 6, 7, 8 mannose, this is the structure. It is approved that this can go to the Golgi for its destination. So this is the process with this. No specific question as such, except the dolicol phosphate, what it does. So now we are looking at the membrane traffic. So we saw that how the protein is synthesized into the ER. Now they are ready to go from the ER to the cis face of the Golgi. This is your cis face of the Golgi, closer to the ER. Then you have the middle stacks. Then you have the trans face of Golgi, which is a distal from the ER. Now, where are the destination? It could be secretory vesicle, secreted outside. It can be membrane proteins going to the membrane. It could be in the lysosome as well. So these are the different destination of the membrane traffic. And also remember that membranes are also trafficked back, like the blue, these blues. Some of the vesicles will actually come out from the Golgi to the ER back. And here the endosomes can be recycled back. So this is the endosome from the plasma membrane. Vesicles are formed. So traffic forward, all the reds, they are called the anterograde transport, anterior grade. Reds are anterograde, whereas the greens and the blues are retrograde transport because they are going back. Although this is going back, but it came from that and going back. So this is also considered as a retrograde. This is anterograde, retrograde, anterograde, retrograde. So these are the different types of transports, and we're going to go into the detail of that. Just shows that from the ER that the vesicle is going to bud and fuse with the target acceptor organelle, like say, and we are going to see that. Cis phase is entry. Then you have the cisternas right here, and the trans phase is exit. These are the secretory vesicle. So ER to Golgi traffic. Just some of the pictures. Now, let's take a look at what are the proteins modified into the Golgi. And this is one of the most important part that make sure that you pay attention. You get as much as 4 set of questions worth of 10 points. So just make sure that you get this one. So the N-linked oligosaccharides that we just saw, they are processed into 2 types out of the Golgi. So processed in 2 types, and where from? Out of Golgi. These are your matured proteins that are going out to the membrane. They're going to the lysosome, and these are different proteins. So one is a complex. So the complex protein has 2 N-acetylglucosamine, 3 mannose, another 3 N-acetylglucosamine, 3 galactose, and 3 sialic acid. So this is complex. You do not have to memorize the numbers and the names of that. Just remember that this is the complex. And only that the high mannose has a large number of mannose and 2 N-acetylglucosamine, whereas this contains an additional N-acetylglucosamine, galactose, as well as sialic acid. But all you have to remember that as complex and high mannose. Now, what we can do as these proteins are modified into complex or high mannose, we can study that how they are trafficked through the Golgi. So let's see that what is happening step by step. So here we are looking at the ER lumen. This was the things: 2 N-acetylglucosamine, 9 mannose, 3 glucose. They are trimmed. All these things are gone. So here you have 2 N-acetylglucosamine and 8 mannose. This will go to the Golgi and trimmed further. This is just only, again, shows that trimming process. And the trimming, if it doesn't happen properly, then this is CNX and CRT. This is calnexin, calreticulin, which we saw all the way down here, calnexin, calreticulin. These are quality control enzymes, and these quality control enzymes will degrade if necessary. And if the proteins are not properly folded or glycosylated, this is lead to this degradation. So here now we are looking at the protein. So let's go from this side. So from here, this came to Golgi. Immediately upon coming to the Golgi, from 8 mannose, 3 of them are reduced, and you have the Golgi with 5 mannose. So this is a distinctive feature that the protein that goes to the Golgi, they will immediately have a 5 mannose at the initial step, whereas that is coming from the ER, or if they're in the ER, they will have 8 mannose. And then in stepwise, they can move through. And then from here, they can be 2 different types. If you add these things, this become a complex protein, or it can be a high mannose protein that we just saw. This is essentially by those modifications. So let's see that what are the things that is happening in the Golgi right here. So see that this is on the other hand, you do not have to memorize these. Here, phosphorylation of oligosaccharide on lysosomal proteins, removal of mannose and N-acetylglucosamine, addition of galactose, addition of N-acetyluramic acid, which is sialic acid. These are the things which are this. See, addition of N-acetylglucosamine, galactose, sialic acid, and NANA is the same. So if you just find these two words, and if it's confusing, remember that this is also known as NANA, N-acetyluramic acid. So sialic acid and NANA is the same. They are the same. All right. So all these proteins, what is shown in here is all these proteins are modified into this in these different compartments. That's basically what has been shown. But again, memorization is not necessary. So here, what happens that we can actually study that where the proteins are in the Golgi and when they have passed the medial Golgi. See that right here, this is your medial Golgi where you have the removal of mannose and addition of the N-acetylglucosamine, galactose, and the complex. We can actually study where in the Golgi our proteins are by some endoglycosidases. So these endoglycosidases, remember that endoglycosidases like ENDO-H, these are some of the enzymes which can remove glycosylation from the proteins. And just remember that as the signal peptidase remove the signal peptide and the proteins get molecular weight, the protein's molecular weight becomes less. When glycosylation is removed by the endoglycosidases, the molecular weight of the proteins get less. So that is the essence. So first, I'll come back to the ENDO-H. First, let's do the ENDO-D. So what question can ENDO-D ask? The ENDO-D is an endoglycosidase which can distinguish proteins whether the protein is in the ER or in the Golgi. How we can distinguish? Remember that in the ER, the protein has 2 N-acetylglucosamine, 8 mannose. As soon as it goes to the Golgi, 3 mannoses are removed and you have 5 mannose. And based on that, we can tell that where is the protein. How? So here in the ER, you have the 8 mannose. In the Golgi, you have 5 mannose. Once you extract the glycoprotein, you treat them with the endoglycosidase and see that the 8 mannose has no effect of the endoglycosidase D. Whereas if you have 5 mannose, this mannose is cleaved from the protein. So this molecular weight of this protein, just remember that just as such, because the molecular weight of this is gone. So the molecular weight of the protein, molecular weight is less. Whereas here, the protein, which is in the ER, with the glycosylation, there is no cleavage. So molecular weight of this protein is more. So how you can do the studies? First of all, you have to put a radio label. So your proteins are radio labeled by 35S methionine. And then you start the translation process and you take samples like, say, for example, time in a minute, 0 minute, then 5, 10, 15, 20, 30, 45, 60. So this is being done with a protein like a VSV glycoprotein, and its transport is seen in 32 Celsius. Whereas this is temperature sensitive. If you make the condition of the cell at 40 degrees, you see no transport. But here, this is the transport that what we are seeing. After 0 minute or 5 minute, this is a band of the protein at the very top right here. Most of the protein has a higher molecular weight because they are in the ER. They cannot be cleaved. Now, which is here difficult unless I say this band is not single one. There is a top band right here, which is getting thinner and thinner, and a bottom band is getting actually, a different color will be helpful. The bottom band is getting thicker and thicker and thicker and thicker. So what it means that the lower molecular weight over the period of time is increasing as the protein is trafficked from the ER to the Golgi. So I will just take those things mark off. But now if you can see that as the time goes by, the top band is getting smaller and smaller. So the higher molecular weight is getting less, and the band at the bottom is getting thicker and thicker. So the lower molecular weight getting more and more over the period of time. So this shows the curve that in the time, if you plot these, percentage of the protein sensitive to endoglycosidase D, that is, and if you plot that, this shows your trafficking curve of that, how the protein is trafficked from the ER to the Golgi. All right. Now what we can do, this is your endo D. Endo H does another traffic, which tells you the question, when the protein crossed the medial Golgi, when? And it can only do it with the complex. It cannot do it with the high mannose. We are going to see that. Only can do with the complex. This is actually very important. It only can do with complex, not high mannose. And we are going to explain you why, and that will be the important question that we are talking about. Multiple questions can be asked from that set of data analysis. So let's take a look at this. We'll skip this one and go to the next one first. So first, what we are seeing in here, these are the proteins which are completely mature. So each word is important. What we mean by the mature protein means that they are done with Golgi, All the modifications are done in the Golgi. So that is what we mean by the mature proteins. So now, the mature proteins could be two different types, and we already saw that, so connect these two. The two mature proteins out of those processed in two types out of Golgi, one is complex, one is high mannose. Complex, high mannose. We are going back to that. Now, what you do that you use endo H, and the lanes are without enzyme, with enzyme, without enzyme, with enzyme. When you take the mature high mannose out of the Golgi, you see that the high mannose, the glycosylation is removed. So that's why the molecular weight is less than this protein. So this means this is sensitive to endo H, sensitive. Whereas if you see the mature proteins are not, they are the same as the original molecular weight, they are resistant to endo H. And based on that, if I tell you that you have a mature protein out of your Golgi, you treated them with the endo H, and you have a protein A, this is protein A, and this is your protein B. Which is what protein? Wherever you see that there is a digestion of that protein, which is sensitive, you know that this is high mannose. High mannose is always susceptible to endo H. But this is complex. So your B protein is complex right here. So your B protein is complex. So even in the mature form, endo H will always digest the glycosylation of the mannose. But if it is with those N-acetylglucosamine, galactose, N-acetyluramic acid, or sialic acid, they become resistant to the endo H. Make sure that you guys actually got this part first correct. Again, remember that I'm going to tell you that you have an A protein and a B protein. Treating them with the endo H, both are matured out of Golgi, and you tell me which protein is what. Whatever is less, you will see that that is high mannose. Whatever is at the same level, which is resistant, that will be complex. This is your data one. So this is from the complex data that I'm saying that 10 points worth of it. This is your data set one. Now, let's go to the slide, which is previous, this slide. So you are going to use the data set one from there to this data set two. This is your data set two. I tell you that this is essentially the movement of the glycoproteins through the Golgi, and you have to tell me that when a protein passed the medial Golgi. So we are giving you these time points, and we are digesting our protein with the endo H, and this is with a protein B. Remember that. This is your protein B from the previous one. So in your data set one, from your data set one, You saw the protein B panel, it was the same. So you concluded that from your data 1 that your protein B is complex. So here, what is happening? The protein B was susceptible to or sensitive to EndoH, sensitive to EndoH, and now it has become resistant and resistant. Why this is happening? If you just look at the graph right here, see that when these proteins in the modification passes the medial Golgi, this is complex, it becomes EndoH resistant. Before that, they were EndoH sensitive, which tells you that the time point in between sensitivity and resistant is when the protein actually passed the medial Golgi. So if we ask you that when this protein actually passed the medial Golgi, or protein B, which is a complex protein, you know that it passed the medial Golgi in between 30 and 45 minutes. Sensitive. As soon as past the medial Golgi, it becomes resistant because the modification was complete for the complex sugars. Remember that this is your data set 2 for only the protein B. Now, we are giving you for the data set 1. So which is not this. So I'm going to draw it out. So I'm just going to draw it out. So here, think about that this will be the panel, the same thing, and the time points and everything is the same. So we have 15, 30, 45, 60. And now what we are seeing that suppose the bands, this is your protein A from the previous data set 1, from your data 1, from the previous one. So your protein A was high mannose. Remember that it is high mannose. Bands will be like this, like this, like this, like this, like this, like this. It will be always at the bottom like this, all throughout. It will not shift upward. And now I ask when the protein A passed the medial Golgi. Most of the people, remember that, most of the people choose that maybe after 60 minutes, it will be modified. But remember that we said in your data set 1, your protein A completely matured was still showing lower band because high mannose is always sensitive to EndoH. So for the high mannose, you cannot do a trafficking study. And if I just give you that data, your answer will be when it passed the medial Golgi, you can't tell. You can only tell for complex. Remember that this is the most important part. You can do the trafficking study for the complex. So why I spend so much time? Because these 4 diagrams can involve you almost like a 10, 12 point of question. So your first data set tells you the mature protein. You identify your A protein high mannose, B protein complex. Then you come to this data. You have again an A panel and the B panel. In the B panel, it's fine. You know that it passed in between these times because the complex is getting resistant after it passing the medial Golgi. For the A panel, because it was a high mannose protein, it will be matured form also after out of the Golgi. It will be always sensitive to EndoH. So you cannot tell the trafficking study. It is not after 60 minutes because you cannot tell. It will never be resistant to EndoH because it's always susceptible. So this is probably the most important question from here. Lots of points. So that's why definitely, if it's needed, scrub it back and rewatch that. I just want you guys to really pay attention to this 2 sets of questions. Very, very important. All right. So this just shows you again the trafficking in between, again tracking through video microscopy. They are going from the ER to the Golgi to the plasma membrane over the period of time. Not that important. So now we are going into a retention signal. After we got the signal, and here we have the retention signal. And it is lysine, aspartic acid, glutamic, and leucine. And in short, 1-letter word, we call this as a KDEL sequence. All right. Now, some of the important things, what happens, that when the vessels are actually formed, these vessels are coated by certain proteins, and we call these codomer proteins. And anything that is an anterograde transport is coated by the COP2. And anyone which is a retrograde transport, they are coded by the COP1, retrograde, COP2, COP1, codomer proteins. Now, what happens that the proteins which are the blue proteins, so for example, these are secretory proteins, no KDEL sequence. Suppose this can go to Golgi, this can go to lysosome, secreted outside. These are needed to be transported out of the ER to the vesicle, to the Golgi, modified, and going wherever they need to go. But unfortunately, what happens that sometimes some of the ER resident proteins, see that they are with the KDEL sequence, they are accidentally, this is the word is very important. It is not purposeful. When the proteins that needed to be transported, accidentally some of the KDEL proteins and the KDEL receptors, which are supposed to be in the ER, also packaged into the vesicle and accidentally transported into the Golgi. But the system does have a mechanism to retrieve those proteins back onto the ER. So they are not lost, they are retrieved back. And we are showing you that how this is possible. So the first thing is the pH in the ER is approximately about 7, approximately almost neutral pH. Where in the Golgi, the pH is approximately 6.8, 6.7, slightly acidic. Even that 0.2 is good, big deal for enzyme interaction. So what happens that at pH 7, all these KDEL soluble proteins, they do not act with the KDEL receptors. Remember that KDEL proteins are all, this is not a single protein. Remember that. All hundreds of different enzymes that is working or resident proteins in the ER, all will have a KDEL sequence. And there are some membrane proteins. These are KDEL receptors. They will bind to any of the proteins which does have a KDEL sequence. But the KDEL receptor at pH 7 does not bind KDEL. So at pH 7, neutral pH, it does not bind to these proteins. This shows close, but this is just like random. See that it's not binding, not binding. On the other hand, what happens when these proteins accidentally are transported at pH 6.8, the KDEL protein and the receptor binds to each other. This becomes high affinity. So again, repeating, at pH 7, KDEL receptor doesn't bind to the KDEL. But the Golgi pH 6.8, the KDEL receptor will bind to the KDEL, and this interaction leads to formation of the COP1 coated vesicle. So this interaction is a signal by itself to form a retrograde vesicle. It's a see that this is an anterograde right here. This is a retrograde vesicle. So this KDEL sequence and the KDEL receptor interaction will form a vessel only with the KDEL proteins, not with the blue ones. They are going somewhere else. They are not binding with this protein. They will go forward. But this will again bind, form into the vesicle with COP1 coated vesicle, and they will be brought back into the ER. And in the pH 7, they will separate out. So one of the question that is asked, what will be the vessel that will essentially coat a vessel? Remember that if it is an anterograde transfer from the ER, no matter what, it will be always COP2. Forward is COP2 and the reverse is COP1. And again, there is always the KDEL or the KDEL receptors. They are accidentally moved into the Golgi and retrieved back. So know the mechanism of this very, very well. All right. So other than X-linked glycosylation, there are some O-linked glycosylation where you have this X-serine threonine and a hydroxyl group is attached with the glycosylation. A couple of these, again, not needed. I did not go into that. This actually tells you the topology, and I told you guys somewhere else that always remember the luminal space is equivalent to the extracellular space. That is the most important part from here. See that the brown dot is in the lumen and the tail is extruded into the cytosol. If this protein becomes a membrane protein, the black part is in the cytosol and the luminal part, which was in the ER lumen, Golgi lumen, vesicle lumen, and when the vesicle essentially fuse with the membrane, the brown part flips towards the outside. So again, we can just tell you that a protein synthesized in an ER, this is how the membrane topology look like. If it becomes a Golgi protein, how it will look like. If this becomes a plasma membrane protein, how it will look like. So definitely remember that. You will see that there are some questions with some pictures in your critical thinking questions. Definitely look at that. And again, remember that, just do that with the plasma membrane. This is the topology in the Golgi. This is the topology in the ER, vesicle, all the same, but it will flip outside to the extracellular space. All right, let's take a look at the membrane traffic from the lysosome, from the transphase of this into the lysosome, and what are the different traffics. So many different, these are all the different proteins. These are acid hydrolases, works with pH. One of the typical things that why the membrane of the lysosome is never digested, because there are glycoproteins with heavy glycosylation, so the proteases and the lipases cannot digest the membrane. You will see some questions in your quizzes as well, equally important. All right, so here we are looking at the whole mechanism, ER to Golgi to the lysosome. And here you have the endocytosis. They come to the lysosome and they can get degraded. And autophagy is recycling of the own organelle when they are run out of function or they just become old and regenerated. All right. How does the cell know which protein to sell in the lysosome? So let's take a look at this. So here, what is happening is that this is a signal. Remember that this is a signal that is a specifically destined or a retention signal for the lysosome. And here we are looking at that how the signals are put to take this protein to the lysosome. See that we have a KDEL sequence from the ER. So what is the lysosomal sequence? So let's take a look at how it is put in there. The lysosomal sequence does not come right from the beginning of the protein like the KDEL sequence. The KDEL sequence is built into the protein, but here the lysosomal proteins does have a glycosylation, which is the signal. But in the protein, there is a signal which helps the enzymes to recognize, to put a lysosomal signal. Again, I'm repeating, the lysosomal proteins does have a signal to be recognized at the Golgi proteins, some of the specific proteins, to put a glycosylation as a signal so that that protein is trafficked to the lysosome. So the first enzyme of that is an N-acetyl. This is how it is read. GLC-NSE is read like N-acetyl-glucosamine. It's read from the first, second half, then N-acetyl-glucosamine. The name of the enzyme is N-acetyl phosphotransferase. And there will be another enzyme, which is phosphodiesterase. Two enzymes are required for putting the lysosomal signal. These proteins are in the Golgi. Remember that. The enzymes which are making these signals are in the Golgi, not in the lysosomes. And they are making proteins so that they can go to the lysosome. So the N-acetyl-glucosamine phosphotransferase has two different sites. One is a recognition site for lysosomal enzymes. And see that these parts, which are in pink, are the recognition sequences. What are the different sequences? You have a dileucine-based sequence, DXXLL or DXL tyrosine-based. And again, you do not have to memorize any of them, but you have a recognition sequence. And then you have a catalytic site, which actually puts some of these residues. So you have a glycosylation already in there. So you have 1, 2 N-acetylglucosamine, and you have 1, 2, 3, 4, 5, 6, 7, 8, 9 mannose in here. And what is happening in here, that it is putting 1 phosphate and N-acetylglucosamine in red onto this protein. And then this phosphate is attached to the mannose 6 place. So again, it's a hexose. And again, I do not have to go into the biochemistry of that, but just remember that how it is done, that this is CH2OH, and the phosphate is right here. And number is like 1, 2, 3, 4, 5, and then a CH2OH. And this is a carbon right here. And this is the 6th carbon. And the phosphate is attached to the 6th carbon of the mannose or the hexose. And that's why we called is an mannose 6-phosphate. The phosphate is attached to the 6th carbon of the mannose sugar, which is a hexose with 6 carbons, like glucose, but again, different side chains. So this is the process where you have 1 N-acetylglucosamine attached with 2 phosphate and 1 uridine. That's why we call it an UDP, uridine diphosphate N-acetylglucosamine. This is the precursor. Remember that. Again, uridine diphosphate N-acetylglucosamine. To each end, there is a sugar. Uridine is a sugar. N-acetylglucosamine is sugar, and that there are 2 phosphates in there. From this, the 1 phosphate and the N-acetylglucosamine, 1 phosphate and the N-acetylglucosamine is put onto the mannose right here. And then the next enzyme, phosphodiesterase, takes off that N-acetylglucosamine. That N-acetylglucosamine, that square, red square, this is gone. Phosphodiesterase takes it one. Only the phosphate remains at the terminal mannose at the 6th base. This is essentially the signal. So just remember that what is happening, again, summarizing, N-acetylglucose phosphotransferase is a cis Golgi enzyme, recognizes the lysosomal enzyme sequences, binds to this, then with the UDP N-acetylglucosamine transfers the phosphate, and phosphodiesterase cleaves the N-acetylglucosamine to leave the phosphate only, which becomes mannose 6-phosphate. So this just shows you the 2 enzymes. So name of the enzyme, both are Golgi resident proteins, and these are the 2 names of the enzyme. So whenever you see this type of picture, you know that these will be your exam question. Which of the following is where? That's basically the type of question we are asking. So very, very important. Sometimes we ask which of the following type of question that we ask, say which of the following type of mutation will lead to a non-functional N-acetylglucophosphotransferase, and what will happen. So suppose that you have this N-acetylglucophosphotransferase and you have a mutation so that it cannot put the mannose 6-phosphate on the protein. And what will happen? If it cannot take the mannose 6-phosphate on the protein, it will not be trafficked into the lysosome. So this type of question we can ask. Again, remember that whatever is the function is to make the signal. If the protein or the enzyme is defective by a mutation, the signal will not be put on and this protein will not go to the Golgi. So this will be a diseased condition. That type of question we can ask. So how this is now trafficked? So now what you have that on the Golgi membrane, this hook-like structure is a mannose 6-phosphate receptor. So the proteins with the mannose 6-phosphate binds to the receptor and it binds to a bud which is clathrin coated. So clathrin coated buds come together and it buds into a vesicle that will form the vesicle and it will go into the lysosome. And a receptor is recycled back and it can take more enzyme into the lysosome. This is basically the mannose 6-phosphate based traffic. All right, so a couple of different storage disease. These are all self-explanatory. Eye cell disease, Tay-Sachs, do not have to memorize. Just remember this. This is what we were talking about, that eye cell disease. N-acetylglucosamine phosphotransfer is absent or mutated. And what happens? there is no mannose 6-phosphate put onto the lysosomal enzymes, and therefore mannose or hydrolases are not phosphorylated. And all of the hydrolases without the signal are secreted outside the cell, because without anything, it just loads onto the vesicle. It doesn't know that it needs to go to the lysosome. It thinks it needs to be secreted out of the cell. It's secreted out of the cell, and severe tissue destruction actually happens. But only just remember that in hepatocytes, which are liver cells, this, even in I cell patients, it demonstrates normal distribution targeting hydrolases. So there may be a trafficking, a different mode of trafficking in those cells as well. So that's one of the important things. So this is, again, I cell and acetylglucosamine. So very important. So definitely questions, definitely remember this. All right. So after that, we are looking at some, these are just that how the vessels are released, TAM, release of histamine from a mast cell, which are allergic response. Your anti-allergy medication, Claritin, Zyrtec, they keep these anti-allergic into the mast cells so that they are not secreted. All right. So after that, we are looking at the endocytic pathways of the lysosomes. You have the phagocytosis, fluid phase endocytosis, autophagy, and they can all come to the late endosome and finally end up in lysosome for the degradation. So these are the different parts. So just remember what is phagocytosis, what is fluid phase endocytosis, what is autophagy, just basic things. Phagocytosis is large particles. Fluid phase endocytosis, as usually also called pinocytosis, is liquid droplets. Autophagy is basically recycling or degrading of older cellular organelle. Yeah, so just to remember those. And phagocytosis is a triggered event. If the bacteria is covered by antibody, it actually helps in the process of phagocytosis much, much faster. So this is just a neutrophil phagocytosing 2 bacteria. You can see that red blood cells are recycled by these macrophages. This is phagocytosis, just example of these. These are all very straightforward. Just pinocytic vesicles form clathrin-coated pits, and these are all very straightforward. So now we are looking at the clathrin-coated pits and the receptor-mediated endocytosis. This is also a form of pinocytosis. So here what you have on the plasma membrane, you have all these cell surface receptor. When it binds to that, if it invaginates, it will form a clathrin-coated vesicle and bring it as an endosome, which will essentially fuse with the lysosome. So we are looking at some of the specific receptor-mediated endocytosis, how the LDL is moved into the blood, because the low-density lipoprotein, they have this cholesterol esters in here, which is the cholesterol fatty acid. It's covered by a phospholipid monolayer. And these are dangerous in the sense that if they burst, all these lipids will essentially stick to the blood vessels and it will cause problems. So to make these things soluble, they are covered by the ApoB protein. And so this is LDL is a lipoprotein complex. And let's see that how the cell actually gets all these phospholipids and the cholesterol esters. Remember that cholesterol esters means is cholesterol versus all the fatty acids that is required for membrane synthesis. So essential part. And again, see that cholesterol esters does have a tail. That is the fatty acids, which make all the different phospholipids. This at top is the phospholipid. These are unesterified cholesterol without the fatty acids. These are the mode of transportation. So the LDL particles, these are formed in the liver, and then they are transported via blood, goes to every cell because all the cells need the lipids and the cholesterol for the membrane biosynthesis, membrane genesis. On the plasma membrane, you have the LDL receptor. LDL receptor specifically binds to the ApoB protein. It doesn't recognize phospholipids. So just remember that. See that the band is ApoB protein, and it only binds to this apolipoprotein B, or ApoB in short, because receptors don't bind to the phospholipids because their structures are floppy, not proper three-dimension. Once they are bound, they find them in these clathrin-coated pits, and these are with the AP2 complexes. These clathrins are grabbing arms that form the vesicle and bring them into the lysosome. Lysosome essentially degrades the apolipoprotein, which gives the amino acid, cholesterol, and the fatty acid, but the receptor is not degraded, the receptor is recycled back onto the plasma membrane to bring those. So this is the process of the LDL uptake. All right. So again, lots of genetic disorder can happen with those. So you can just expect some of the related type of question. And then this is the most important part in here, that what are the different fates of the cell surface receptors for endocytosis? Remember, one is recycling, one is degradation, one is transcytosis. So we are looking at each one of them. So that is very important. So here we are looking at first recycling of the receptor at the same domain. What it means by this, that the LDL receptor was on the plasma membrane, that is recycled to the same plasma membrane. There is no degradation of this. This is your first recycling. Just remember that this was the first one. Number two is the transferrin cycle. So know this transferrin cycle very well. This is also a question that is missed by a lot of people. So know this one very well. So what is happening, that transferrin is a molecule which is released into the intestine by the cell, and it brings absorbed iron into the cell for lots of different functions. So the electron transport chain requires a lot of iron and other hemoglobin and everything. So what happens is that this apotransferrin is without any iron, that is released into the intestinal lumen where you have the pH 7. And it binds to the iron, picks up 2 of the iron. Once it picks up the iron, it binds to the transferrin receptor tightly, and it's endocytosed. So transferrin receptor binds to that, and it is endocytosed. Now it goes to the endosome. In the endosome, the pH drops to pH 5, and the iron is actually released from the ferrotransferrin. And now it becomes apotransferrin. This apotransferrin now goes through the vesicle and recycled back with the receptor all the way into the plasma membrane. And now at pH 7, the apotransferrin actually is released from the receptor to catch more iron. This is the entire cycle. So what is important in here? When the iron-bound ferrotransferrin has a high affinity, at neutral pH to the transferrin receptors. On the other hand, the apotransferrin has a high affinity binding to an acidic pH. See, apotransferrin binds at acidic pH. Fe plus ferrotransferrin binds at neutral pH. This is neutral. This is acidic. But in neutral pH, apotransferrin dissociate from the receptor. As soon as this pH 5, 6 goes out or exocytosed, the cell environment is pH 7, apotransferrin is released. And now if it binds to the iron, it becomes ferrotransferrin, and now it will bind back to the transferrin receptor and recycle back. So a typical hypothetical question that we asked is like, suppose you have a patient where the transferrin receptor cannot sense the pH, neutral pH, and what will happen at that time? So this is fine. So in that case, if it cannot sense the neutral pH, that means that apotransferrin will remain bound in there, and apotransferrin will not be released. So that person will not be able to absorb iron as efficiently and bring it back, and probably suffer from the iron deficiency. That's actually happens. This is a genetic disorder that can happen. Again, remember that just a mutation where the transferrin receptor cannot sense the neutral pH, where it can dissociate from the apotransferrin. It keeps it so that it cannot be released to get new iron molecules. Know the content very well. Acidic pH, apotransferrin binds. Neutral pH, ferrotransferrin binds. And in neutral pH, apotransferrin is released in a normal condition. But in a disease condition, if there is a mutation where it cannot sense the neutral pH, then the apotransferrin will not be released. So just remember that what are the possibilities. All right. So the recycling of those two, LDL and transferrin, then we are looking at a degradation. Here, an EGF, epidermal growth factor, this signal is brought into the cell. And here, the signal as well as the receptor, both are degraded. So degradation of after endocytosis. The previous one, none of the receptors were degraded. They were recycled. Here, it is degraded. This is an extra measurement for some of the intercellular signals, which are mainly for growth control, so that these are some anti-cancer measure, so that the receptors cannot bind to stray growth factor molecules. That's why this is done. But it is degradation of the receptor as well as the material after traffic. Then you have a transcytosis. The transcytosis happens in the intestinal wall. This is an example of that. And a classic example that how the babies actually can receive the antibodies from the mother's milk, essentially after birth, while their immune system are still developing. So the antibody molecules, the IgG, this is from the mother's milk, and the neonate ingests that. The antibodies are heavily glycosylated and they pass through the intestine without being digested. They are not going to get digested. They will be intact. And the lumen of a neonate intestine is about pH 6. In the pH 6, the antibodies have a high affinity for the antibody receptors. So you can see that. And this will end up in a clathrin-coated vesicle. And the antibody is going to be trafficked from the apical surface. Remember that this is a part of the intestine, and we are looking at just that. This is the hollow of the intestine, and this is the blood side. So it is going to be trafficked on the blood side, where you have a pH 7 outside towards the blood side. And here it becomes low affinity with the receptor, and that's why it releases the antibody on the blood side. So again, what we mean by this high affinity or low affinity, high affinity means that the receptor binds to the antibody to bring it inside the cell. When it goes endocytosed and transcytosed, why we call it transcytosis? Because it is transcytosed from the apical surface to the basal surface. It's traveling transcytosis from apical to basal. When it's going to the basal surface, so see that this is your luminal membrane, which is similar to the apical membrane. Remember that, apical, luminal / apical. And it becomes low affinity, which releases the antibody into the blood of the neonate, and protect. So this is your transcytosis. Again, what are the different fates? Remember that we saw this slide in here, and these are the three things. Remember that this will by itself, this is a question. What are the fate of the cell surface receptors after endocytosis? Recycling. We saw LDL, transferrin. We saw degradation, EGF receptor, transcytosis, antibody molecules. These are the three examples of that where they can go. All right. So after that, we are looking at that how the things are trafficked, molecular mechanism of traffic, and how this happens. So the first thing is that what we are looking at is the mechanical force. First is the mechanical force for the trafficking. And this mechanical force of formation of the vesicle or pinching of the vesicle. Think about that. You can use a finger to make, suppose you have a balloon which is not very tightly packed, and then you can just grab that balloon and make a smaller structure out of that. So this grabbing mechanism is basically what we call as a mechanical force. It could be clathrin coated, COP1 coated, or COP2 coated. These are the three mechanical force for formation of the vesicles. So let's take a look at that. Where are what? So if you see that anything that goes from the ER to the Golgi forward, they are COP2 coated. Anything that is retrograde, that is coming from Golgi to ER, or in between Golgi, retrograde. Anything retrograde from the Golgi is COP1. And everything else, anything forward from Golgi or anything forward from or inward, rather, endocytosis from the membrane is clathrin. So it's pretty easy to remember. Again, remember, anterograde from ER, COP2, retrograde from the Golgi, COP1, and anything forward anterograde or retrograde from Golgi or retrograde from the membrane is essentially coated by the clathrin. And again, these are the mechanical forces for forming. So these are called the triskelion structures. They are like this, and they just grab the membrane to form these vesicles. So this is the mechanical force of the clathrin, and it just shows that what happens. This is a mechanical force. So now, after the mechanical force, there is also another thing, is where is what is going. And these are specific adaptin molecules that's going to take them to the different parts, and that binds to specific receptors. So remember that the adaptins binds to specific receptors, which are again covered by the clathrin molecules. And definitely for this, I will highly suggest to look at the supplementary videos. Like these are YouTube third-party videos that how these are formed. That's very important as well. So here, the protein signals for the adaptin binding. So we are looking at that how these bindings actually are taking place. This is a plasma membrane receptors. So if any cargo is bound to a plasma membrane receptor, the receptor has a following sequence: phenylalanine, arginine, X tyrosine. You do not have to memorize, but it is just a sequence like this that binds to a specific adaptin molecules, and this adaptin molecules will bind to the clathrin, and this will go to the endosome. On the other hand, if there is a mannose 6-phosphate, this was the mannose 6-phosphate receptor, and they have a sequence of phosphorylated amino acid. When there is a separate adaptin molecule that recognizes that phosphorylated amino acids, and when this vesicle will be formed, this will go to the lysosome. This is how the traffic is destined. This is a specific adaptin from cargo molecules coming from plasma membrane surface to the endosome. This is going to the lysosome. Different adaptin molecules. So this is very, very well. So definitely do remember that this is recognition. Where? So that's basically very important molecule. So the mechanical force is essentially the clathrin COP1, COP2, and this is some of the destination or the address. And we are going to see more of these. So this shows you that how a vesicle is pinched off. One of the important protein is here called a dynamin that works with the GTP, responsible for vesicle pinching after hydrolysis of GTP. So definitely this is just this work. Like the question is, which of the following protein is responsible for pinching of the plasma membrane by the hydrolysis of GTP? Once the vesicle is fully formed, is dynamin GTP. Dynamin, non-dynactin, non-dynamin, non-dynin, but dynamin GTP. So just make sure that you remember this one very well. Very specific question. Star marked questions. So these are again COP1 intra-Golgi, Golgi retrograde, and COP1 ER2 Golgi anterograde traffic. We already talked about that. Then we're going to talk about some of the GTPases. These GTPases are small molecules, and these are very, very important for recruiting some of these cotomer proteins, COP1 and COP2. So we already saw these adaptins recruit the clathrins. Remember that? These clathrins are recruited by the adaptins. Now the COP1 and COP2, right here, COP1, COP2, they are recruited by these GTPases. And what are these? See that COP1, these associated GTPases are SAR1, COP1 and clathrin, they are all with these ARFs. And what exactly they do? Remember that the SAR1 and ARF GTPases are involved in the vesicle budding by vesicle coat recruitment. And let's see that how it works. So here, suppose this is an ER lumen, and you have a COP2 that will take this, make a vesicle and take it to the Golgi. So here you have these SAR1, which is associated with an GDP, and you have a tail right here, which is kind of like tucked into. This is a soluble GTPase protein. And how it works, the GTPase, and remember that, how the GTPase works, when a GTPase is bound to a GTP, it's an inactive state. Now, there is another protein which is called a guanine nucleotide replacing protein, releasing protein, guanine nucleotide exchange factor, replaces the GTP with a GTP. The GTP bound GTPase is active. After its function is done, there's another protein called GAP, which is a GTPase activating protein. This will go into an inactive form, basically. So this is essentially the cycle that goes on. So let's see that what is happening. This is an inactive GTP bound SAR1. Now this SEC12, this is your guanine nucleotide exchange factor, which replaces the GTP with a GTP. Now the GTP bound SAR1 extends that tail, which is hydrophobic N-terminus, and it inserts onto the membrane. And this SAR1 will be used as a scaffold to recruit these COP2 components. Let's see how it happens. So the first picture is the previous part, and see that SEC23 and SEC24, these are the part of COP2 parts, which are the mechanical force. Remember that we are still talking about the mechanical force. So these are the proteins, SAR1, which is inserted into the membrane. This is your ER membrane. And these are the vessels, which are the cargoes right here, which are the proteins that are going somewhere. And the SEC2344, these are the green ones, which are recruited onto these SAR1. And that helps the pinching of the vesicle. Again, remember that, as I said, COP1, COP2, and clathrin, these are mechanical forces, and SAR1 is helping in that process. As soon as the vesicle is pinched off, the GTP is hydrolyzed right here, and see that they have become green. And as soon as they become green, they will dissociate from the membrane, and the whole of the COP2 vesicle will fall apart. And you have an uncoated vesicle. And this uncoated vesicle will go and dock onto the destination. Now, the question is that how you will recognize that destination. We'll come to that in a second. So this is all about the specificity of packaging and all those. So here also another one. This is an ARF. This works with COP1 and clathrin. So instead of a protein tail, this has a fatty acid tail. So this is a lipoprotein bound with a GDP, but same function, guanine nucleotide releasing protein. GDP is replaced by the GTP. As soon as the GTP is replaced, the G is added to that, the fatty acid tail inserts into the membrane. And now on top of that, the same way like this, the COP1 and clathrin can form and pinch off the vesicle. So these are all mechanical forces for this formation. So this, you can see that this is the cotomar proteins active, and this cotomar coated vesicles is formed, all mechanical forces. All right. So mechanism of vesicles formed, we just saw that. What are the molecular signal of the cause bind to only appropriate tolerate? This is the membrane specificity, and how to transport vesicles to their target organelles actually fuse. So we are going to talk about this. So for this, the proteins that are involved are called snare. So snare is a SNAP receptor and RAB GTPases, and they play a role in the vesicle traffic and specificity and the fusion. So what happens? The donor from where the vesicles are formed, see that these are your COP1 or COP2 or clathrin, and here you have a vesicle snare, V snare, that goes all the way to the target component, and the target does have a T snare or the target snare. These things interact each other and fuse the vesicles that we're going to see. But these are the mechanical forces of fusion. Again, I'm repeating that. Mechanical force for formation of the vesicles are formed by the SAR1, ARFs, and then you have the clathrin, COP, and the COP1 and the COP2s. Those are formation of the vesicle. Fusion of the vesicle will require T snare and the V snare. Recognition of the particle will need the RABs. And I'll sum it up so that it becomes easier. What are the RABs? We're going to see that. This just shows you all the independent components of that. This is talking. We'll just show you how it's fused. So what we are going to see is that the transport vesicle, has a RAB. This is a RAB GTP. These are small molecule GTP as well. This is the address. This is essentially the address. Think about that. This is the letter on the letter, and you have the address and the RAD effector. This is your mailbox in front of your house with the house number written on that. And they interact so that they are at the proper destination. And the VAMP is VAMP is a specific name. This is the vesicle snare. Syntax in SNAP 25, they are specific names. They are T snares. Individual names are not required. V snare, T snare, good enough. These are the mechanical forces, again, mechanical forces for fusion. We talked about the mechanical forces of formation of the vesicle. This is for the fusion, V snare and T snare. They will interact and basically bring down the vesicle together. So let's take a look at that, what happens. So these are, again, RAB GTPs is a revolving vesicle docking with the destination or the target. It is the address. 150 different types. Do not have to memorize that. Just only what are the RAB GTPases. Remember that SAR1 and ARFs, those are the GTPases which recruit the mechanical forces for pinching of the vesicle. RAB GTPases help in the destination. So now what you are seeing that the syntaxin and the SNAP 25, they are going to coil together and bring this vesicle down, and they will essentially fuse. They will bring down and they will fuse. In that process, what's going to happen, both of these molecules are going to wrap up like this. And then at the last step after the fusion, the V snare and the T snare are recycled back by this protein called the NSF. And how this works, the NSF is a hexamer with an alpha snap. And with the help of an ATP, it sits on top of that, untangles the V snare and the T snare, and the V snare is recycled back. So again, what is the question we ask from here? Is which of the following is the last step? Which of the following is the last step in vesicular transport of protein requires hydrolysis of the ATP? Again, remember that. Again, repeating that. Which of the following steps in the vesicular transport of protein requires hydrolysis of ATP? If the disassembly of the snare complex is the last part with the help of the NSF, not NSP. This is NSF. Just make sure that the typo here, I will correct. This is NSF and the alpha snap. So this is the overall process of that. Now, this is the overview of that. But I just want to take just a little moment just to summarize that part that where people get confused with that. So the part A is vesicle formation, vesicle form. I'm just writing formation, vesicle formation, and B is fusion. B is fusion. So for each one of them, there are several things that happen. Number one, what is packaged in the vesicle? What is packaged? So a couple of examples of that. We got this one for the adaptin. Remember that? Adaptin receptor complex. These are the specificity. Adaptin, receptors, plasma membrane, and of those. These are packaged the same thing. Number two, what are the mechanical forces of the formation? Mechanical force. Mech force. Mechanical forces coming from first the SAR1, depending upon that, plus ARF, and B coming from clathrin, and plus COP1 plus COP2. So these are essentially all it's needed. What is packaged by adaptin and the receptor? specific mechanical force coming from SAR1, ARF1, and clathrin. So know this. After that vesicle is formed is the fusion. Right here, the number 1 is where going. Address. /address. ADDRESS. Address. This is RAB GTPases. All these different types, RAB GTPases plus RAB effector. So just think about that. RAB GTPase is in the vesicle. RAB effector is at the destination. This is the destination recognition. This is on the vesicle. So the vesicle RAB GTPase must be recognized by the RAB effector. Number 2, the second part is the vesicle fusion. So how the vesicle is essentially fused, it is the V snare plus T snare. They coil each other and pull the vesicle down to fuse it. And then ultimately the NSF plus alpha snap, they actually recycle the V snare and the T snare back with the help of the ATP. So this is the entire process that is in there. The only thing that we did not talk that how it goes from point A to point B, it just does not diffuse out. That is when we go to the cytoskeleton and the motor proteins, we're going to talk about that. So that will pretty much do it, I guess. Just make sure that know all your practice quiz question, a critical thinking question. I did not post any discussion posts, so that part is that. But just make sure that you look at the supplementary videos. That will also help. And if you just look at these particular slides, you guys should be all good.